A very good morning to all of you. Professor Chantala Ratnasekara, Vice Chancellor of University of Sri Lanka, Honorable Resource Persons, Professor Julian Hanley, Division of Animal and Dairy Science, College of Agriculture, Qtn National University, South Korea, Professor G.L.L.P. Silva, Department of Animal Science, University of Peradeniya, Sri Lanka, Professor Suranga Koditwaku, Department of Animal Science, University of Peradeniya, Sri Lanka.
Professor Indunil Patirana, Department of Animal Science, University of Ruhuna, Sri Lanka. Dr. P.G. Senaviratna, Director of Animal Breeding, Department of Animal Production and Health, Sri Lanka. Dr. Hiruni Vijayasena, United States Department of Agriculture, Meat Animal Research Center, United States of America.
Professor Saman Herat, Dean, Faculty of Animal Science and Export Agriculture, respected Deans of the Associate Faculties of Ovulation University, Dr. Deshani Mudannayaka, Head, Department of Animal Science, respected Department Heads of Ovulation University, Directors of the Centers, Mr. Nuan Lianage, Project Coordinator, ELTA is a Head TP of the Department of Animal Science.
Senior lecturers, and all members of the academic staff, graduates and undergraduates from fellow state universities, experts of the sector, industrial leaders, NGOs, policymakers, UW alumni, entrepreneurs, committee members, graduates and undergraduates of Ovulation University who are present here with us today. It's a pleasure to welcome you all into Farm Animal Breeding and Reproduction Forum 2022. Given the current circumstances, the Department of Animal Science had taken tentative measures in organizing this annual event for the first time in a virtual format to safeguard the health and safety of all event attendees and to adhere to the state-mandated national health norms.
The forum is therefore conducted via Zoom and will also be streamed live on the official YouTube channel and Facebook page of the Animal Science Society. All queries posed through Zoom, Facebook, and YouTube regarding matters taken into review will be addressed during the panel discussion. We would most kindly appreciate it if all participants could contribute to the quality of the program by muting yourself when not sharing and preventing any interference during the day's proceedings.
The productivity of the livestock and poultry industries is dependent on the utilization of appropriate breeds for the various environments and agricultural systems in which they are maintained, as well as correct nutrition management to achieve optimal reproductive results. The development of the livestock and poultry industries in Sri Lanka is a top priority for the government. However, the livestock and poultry industry's contribution to the national economy is insufficient, underscoring the sector's need for improvement. The recognition of the need of separate expert discussions and knowledge sharing in order to close the gaps in Sri Lanka's cattle and poultry industries had led Ovulation University to give way to Farm Animal Breeding and Reproduction Forum 2022.
The forum will hence be conducted under the theme Potentials and Challenges of the Farm Animal Breeding and Reproduction, Sri Lanka and the World. It is organized by the Department of Animal Science in collaboration with ELTA as a head TP of Department of Animal Science and the Animal Science Society. The principal aim of the forum will be to discuss the present status of the genetic potential of Sri Lankan farm animals, a mechanism to utilize the existing gene pool for getting maximum productivity, and global trends on enhancing the productivity of the livestock and poultry industry.
Local and foreign farm animal breeding and reproduction experts, policymakers, research institutes, university students, UW alumni, industry experts, and entrepreneurs who have graced the event will take part in this important forum and share their insights and perspectives on the subjects being addressed today.
For the joining of the event, it's with great honor I invite Dr. Deshani Mudannayaka, Head Department of Animal Science, to welcome the gathering and brief the objectives of the forum on behalf of the organizing committee.
Thank you, Rasika. Very good morning to all of you. It is my great pleasure to welcome all the participants and distinguished speakers to the Farm Animal Breeding and Reproduction Forum 2022, which is held for the first time by the Department of Animal Science, Ovulation University of Sri Lanka. First of all, I would like to remind Professor Chantala Ratnasekara, Honorable Vice Chancellor, Ovulation University of Sri Lanka, Even though he could not join with us today due to his very tight schedules, he extended his compliments and best wishes for the first ever Farm Animal Breeding and Reproduction Forum 2022, which is organized by the Department of Animal Science. Then I would like to warmly welcome Professor Saman Herath, Dean, Faculty of Animal Science and Export Agriculture, who has been a strong pillar of support behind all the activities we conduct at the department as well as the faculty. Thank you for being with us, sir. Then it is great pleasure to warmly welcome the most distinguished guest speakers to today's forum who join in with us live from various international and national institutes for the Farm Animal Breeding and Reproduction Forum 2022. So I would like to welcome Professor Pradeepa Silva, Senior Professor, Department of Animal Science, University of Peradeniya, Sri Lanka. Dr. P.G. Seneviratna, Director, Animal Breeding Division, Department of Animal Production and Health, Sri Lanka. Professor Suranga Kodituaku, Professor in Department of Animal Science, University of Peradeniya, Sri Lanka. Then Professor Jun Hyun Lee, Professor in Chunnam National University, South Korea. Then also Dr. Hiruni Vijayasena, Postdoctoral Research Fellow, United States Department of Agriculture, Meat Animal Research Center, USA, and Professor Indunil Patirana, Professor in Department of Animal Science, University of Ruhuna, Sri Lanka. Thank you very much for joining, accepting our invitation and joining with us live among your very busy schedules in this special morning for the Farm Animal Breeding and Reproduction Forum 2022. So in this special morning, I would also like to welcome all the academics of the Department of Animal Science and Uvavelasa University and every other personalities representing the private, public and non-government organizations, and also my dear undergraduate students joined with us today. So every year as the Department of Animal Science, we held special forum to discuss various issues and potentials with regard to the animal science and technology. So over the last few years, we have held few such forums, actually many such forums, such as Aqua Forum, Poultry Forum, Dairy Forum, and etc. So these were able to make our undergraduates and stakeholders very aware about the current status, and also it opened up the new inspiration sessions. So in this year, we decided to hold the first ever Farm Animal Breeding and Reproduction Forum in order to make aware the undergraduates and our stakeholders. So this was actually initiated and incepted by two wonderful members of the department, that is none other than Dr. Prabudha Manjula and Dr. Sudhini Fernando, the senior lecturers of the department. I should thank them to initiate this interesting forum, which became a success today. So this is held with the participation of diverse audience covering the entire livestock and poultry industry in the country, universities and private partners and students who are interested in animal genetics and breeding, and to discuss the current status of and understanding of genetic resources and potential benefits to be gained by accessing our unique genetic resources in the country. So today, the forum will be held with the theme of potentials and challenges for farm animal breeding and reproduction in Sri Lanka and the globe. The key objectives of the forum are to summarize the present status of the farm animal breeding and reproduction in the country, and to explore the recent advanced techniques and recent research on the farm animal breeding and reproduction in the world, and to foresee the ways of improving the farm animal breeding in the country. So I'm sure this will create an invaluable communication channel with the academics, industry, and policymakers, and also, most importantly, with the students for sustaining the growth of the livestock and poultry sector of future in Sri Lanka. So with that, I would like to stop, and I wish you all a very fruitful and interesting session. Thank you very much.
Thank you very much, Madam. Now, I would like to invite the Dean, Faculty of Animal Science and Export Agriculture, Professor Saman Herath, to share his most valued thoughts with us.
Yeah, very good morning, everybody. I hope you can hear me. Yeah, all the invited speakers and the resource personnel, Dr. Mudana, head of the Department of Animal Science, and all colleagues representing the Faculty of Animal Science and Export Agriculture, and more importantly, our students and all the participants and the well-wishers. So let me congratulate the first or the maiden forum on farm animal breeding and reproduction, which is organized by the Department of Animal Science for the first time, the history of the faculty and the university. So I hope this will be a remarkable day to discuss the matters, issues, and also the potentials related to the farm animal breeding and reproduction of the sector. And also I take this opportunity to welcome all the invited speakers and also the resource personnel on behalf of the Faculty of Animal Science and Export Agriculture and also for Ovulation University. So for those who are joining with us for the first time, I just thought of giving a brief introduction about the university and this great movement. Actually, we have a vision to be the center of excellence for value addition to the national resources of Sri Lanka. So in this journey, we have paid attention on two important aspects. One is promoting value addition to the national resources. So under that, when it comes to us, we focus on agricultural and related resources. The second important thing is promoting entrepreneurship. Actually, these two concepts have been inculcated in our all degree programs because we want to provide the basic information and all practical aspects related to these to our students. So our belief is that our students will be the leaders in future who could do a significant contribution to the national economy or the national development. So under that, the other important thing is as a university, so we have committed staff in our university and at our faculty. They have been given another 3 tasks to serve for the whole world. One is teaching and mentoring, which is the main task of our all teachers or the lecturers. And the second important thing is doing research, focusing to the value addition of the natural resources of the country. The third important thing is engaging in community services, development activities. So this will be one of those, and I hope this will be definitely a forum or a platform to discuss, as I told before, the issues, the problems and the matters and also the potentials that we should think of for the betterment of the country and the world. So as a country, we have a significant contribution in agriculture and also in livestock, this animal husbandry area. My thinking or my thought is we should pay more attention on this animal production and breeding because we mainly focus on the management of the animal husbandry, but our attention has been paid very less towards the farm animal breeding and animal production or reproduction. Therefore, this will be a base, this will be an initiation made by the Department of Animal Science to discuss or to bring all expertise people into one forum and collecting information and taking them into a national drive later. Therefore, I would like to make a suggestion to the organizers and also these our lecturers to have a write-up or have a kind of compiling document after the great speaker, after having all this information at the end of the day. So this will be very important. And as a faculty, we can take it forward to the relevant people later on. That is my suggestion, and I don't want to take much time because there's a lineup. There are so many people who are prepared to talk in this great event, representing both local and foreign, platforms. So we should give them to talk. So that is the most important thing in this, the forum. So I appreciate again the organizers and also the head of the department and all colleagues, so who have, made a great effort to organize this event for the first time. So this will be a wonderful event, definitely. And also I appreciate all the resource personnel and the invited speakers who have joined with us, extending your support to us. So this will be very important in this great, event. So that is appreciated, and all the participants, including our students. So you are there. So hope you will get a lot from this great event at the end. So I wish the success. I hope it will happen, and thank you very much for inviting me and giving an opportunity to talk in this great event. Thank you all.
Thank you very much, sir. And once again, as head of the department mentioned the main objectives of the forum, I would like to brief once again about the forum, for which will be held for the first time in the university history, the Farm Animal Breeding and Reproduction Forum 2022. So in today's country, the productivity of the livestock and poultry industries is dependent on the utilization of appropriate breeds for the various environments and agricultural systems in which they are maintained, as well as correct nutrition management to achieve optimal reproductive results. The development of the livestock and poultry industries in Sri Lanka is a top priority for the government. However, the livestock and poultry industry's contribution to the national economy is insufficient, underscoring the sector's need for improvement. The recognition of this need to separate expert discussion and knowledge sharing in order to close the gaps in Sri Lanka's cattle and poultry industries had led Ovulation University to give way to Farm Animal Breeding and Reproduction Forum 2022. So the forum will be conducted under the theme Potentials and Challenges for the farm animal breeding and reproduction, not only in Sri Lanka, but also in the concept of the world. It is organized by the Department of Animal Science in collaboration with Elta Elzahead VP of Department of Animal Science and the Animal Science Society. The principal aim of this forum is to discuss the present status of the genetic potential of Sri Lankan farm animals, a mechanism to utilize the existing gene pool for getting maximum productivity, and global trends on enhancing the productivity of the livestock and poultry industry. Local and foreign farm animal breeding and reproduction experts, policymakers, research institutes, university students, UWU alumni, industry experts, and entrepreneurs who have raised the event will take part in this important forum and share their insights and perspectives on the subjects being addressed today. And it is with deference I invite Dr. Prabodha Manjula to chair the forum and steer ahead the day's proceedings.
It's my great pleasure and honor to introduce the distinguished resource persons of the first animal breeding and reproduction. First of all, let me introduce the first speakers of the forum, Professor Julian Hanley, Department of Animal Science, Faculty of Agriculture, University of Pedigree, Sri Lanka. Professor Pradipha Silva is a senior professor in the Department of Animal Science, University of Pedigree. She holds bachelor degrees in agriculture from University of Pedigree and pursued her PhD in animal genetics and breeding from University of Sydney, Australia. Professor Silva is an animal geneticists with special reference to the population genetics, molecular genetics, and breeding systems of indigenous animals. Her expertise covers the area of animal population analysis and biodiversity evaluations using both quantitative and molecular approaches. And she involved in several international multi-country research projects as well as national research projects. Mentioned, she did the comparative analysis of climate resilient biodiversity of home garden ecosystems in different agroecological regions of Sri Lanka. So a national thematic research funded by the National Science Foundation starting from last year. And also genetic variations on the control of resilience of infection disease in small ruminants for improving animal productivity. Multi-country projects funded by the International Atomic Energy Agency. And also, She is involved in development and applications of decision supports to conserve and sustainable use of genetic diversity, indigenous livestock, and wild relatives. So multi-country projects funded by the Global Environment Facility through the International Livestock Research Institute, Pedigree in 2009 to 2000. Not last but least, she has been contributing to various national development activities by holding membership in different national bodies. She is the project leader in comparative analysis of climate resilience, biodiversity, and home garden ecosystems, and also the principal investigator for the genetic variations on the control resilience and infection disease in small ruminant with animal productivity. And also, she is worked as national project director of the development and application of decision support tool to conserve sustainable use of the genetic diversity of indigenous livestock and wild relatives. And also, she contributed through memberships by holding the members of the National Invasive Species Specialized Group of Ministry of Environment from 2013 to today. And also, she is the member of the National Committee on Agriculture Biotechnology of the Sri Lankan Council for Agriculture Disease Policy, 2009 to today. And also, she is the member of the National Animal Breeding Committee of the Ministry of Agriculture and Livestock from 1980 to today. And also, she is a member of the National Consultative Committee for the preparations of first reports on the state of the world's animal genetic resource from year 2000. In addition, she was a member of the Technical Evaluation Committee on semen importations of the Department of Animal Production. Also, a resource person for curriculum development for the Diploma Agriculture Program at the Technical College, National Institute of Technical Education of Sri Lanka, 2002. Also, she is the member of the Task Force number 5, Impacts in the Biodiversity Committee appointed for the completions and involvement of the biodiversity conservation of Sri Lanka and framework for actions, natural resource management, biodiversity divisions of Ministry of Environment and Natural Resources. So I would like to warmly welcome Professor Qtn Silva from the University of Peradeniya, and she will going to talk about the present status of animal genetic resources in Sri Lanka. Since Professor Silva is still the base to the program, so we'd like to welcome take the chance to welcome the second speakers of the day. We will be back to the Professor Silva, and she is joining us. So, since let me introduce the most distinguished speakers of the day.
Am I audible now?
Yes, madam. You can hear.
Okay. Yeah, sorry about the slight delay because it was so unexpected. So I was ready by 9:30. Yeah. Okay. Shall I start now? Or?
Yes, madam.
Okay. Okay. Right. Can you see my screen?
Yes, we can see.
Right. Good. Okay. So I'm supposed to deliver this important topic today. I'm very happy that I was given this chance, and thank you very much for the organizers, especially Dr. Sudhini, who hunted behind me several times on this important event. Okay, so I will keep it short because I have to keep the time. The present status of animal genetic resources in Sri Lanka. So my presentation lineup is this. I will talk about the profile of animal genetic resources and then move into status of genetic resources of different species. And then I will talk a little bit about wild relatives as well, because otherwise it is not complete to talk about genetic resources without the wild relatives. And then I will move on to very short concluding remarks. So I will introduce the profile of animal genetic resources with the context of the country's profile. So you are familiar with these two maps, I guess, because we are very fond of this diversity that we are having, especially the agroecological zone diversity. We have 46 different agroecological zones, and we talk about the diversity of these the zones. And also we talk about that is the natural diversity, and we talk about the man involvement, how we use the land, and how we generate the land cover with the involvement of human beings. So you may be wondering why under genetic resources I am talking about these two, because these two aspects are helping us to preserve our animal genetic resources, and also they are sort of fostering the animal genetic resources in the country. So therefore, with this vast diversity, we have varying farming system, varying animal systems, and varying resource levels and diversity that foster the diversity of genetic resources. And also because of the different land use patterns, we have we have a fortune of having blend of different genotypes, and also the location of the country is also a matter, also helping there in getting the different blends of genetic resources. So having said that, let's move on to a very introductory slide that I have when we talk about genetic resources. Before I move on to the domestic genetic resources, I should introduce this profile as well, because we have very rich faunal diversity. If you can see this, the mammalian species, bird species, we are very rich in reptile, amphibian species, yet to find some of the species that we are having in our surrounding. And this vast diversity is sort of fostered by forest coverage, which is 29%, and we are sort of heading into 32% according to our targets. And we have out of this forest coverage, we have 26.5% protected areas. We are providing shelter to these wild animal genetic resources. We have moving to domestic animal genetic resources. So I'm basically covering these mammals, avian species that we have in the under domestication. The red color typing will indicate, sorry about that. Red color typing will indicate the leading species that we have under domestication, and under avian species we have chicken as the dominating species. And all the other in the listing here, the species are also adding to genetic resources that we are having in our country in a small manner. And also, other than these main species, we have different rodents, reptiles, invertebrates, fish, crustaceans, mollusks, adding to when you talk about broad area of animal genetic resources. Okay, so I have a very fade the color cats and dogs. We have a good resource there as well when you talk about when we talk about the broad sense, in broad sense, the genetic resources. Okay, so if we try to categorize the genetic resources in the country, the prominent domestic genetic resources that we can categorize into exotic and indigenous in a broad sense. And when you say exotic, we are having continually imported genetic resources like Frisian, Jersey, Sahiwal, when you talk about cattle breeds, and Jamnapari, Saanen like goat breeds. So we are continually importing those exotic genotypes. And in indigenous category, we have native individuals as well as adopted individuals, which have been imported earlier, and they have sort of domesticated and adapted to the native environment. We talk about these different type of genetic resources under these different species, subsectors: dairy, swine, poultry, goat, and sheep. Okay. So let me take you through the distribution of these the genetic resources we have in the country under these different species. And when we talk about the distribution, we cannot forget about the climatic conditions in our country. So the distribution of these genetic resources are manipulated by the climatic zones. So these are not agroecological zones. So normally when the genetic resources are considered, we talk about the climatic zones, agroclimatic zones. There are 7 of those, and the distribution is determined by these agroclimatic zones as well as the farming system in these agroclimatic zones, because the resource base is determined by these 2. Okay, so let's move on to the overall distribution of the animal populations in the country. The maps I am showing are with the courtesy of Department of Animal Production and Health, the entity which is sort of tracking the populations at the moment. So the graph will, the map will give you the shades of the map, not the pie charts, will give you the poultry populations, where you will find that the Western province is dominating, and the pie charts will give you the composition of other livestock populations, like the yellow color for cattle, green color for buffalo, and so on. Okay, so you will find that these areas are densely populated by the animal species, the genetic resources that we are talking about, and some areas are sparsely populated by the animal genetic resources in the country. Okay, going by a species. Now let me talk about cattle animal genetic resources. So we have basically 3 genotypes: Indian breeds and their crosses. Here it is Indian breeds and their crosses, European breeds and their crosses, and the local. So this is the composition at the moment. We have 24% local type of animals, 33% Indian breeds and their crosses, and 43% European and their crosses. Okay, so the map will give you with the shades the populations as a total. If we go by the locations, actually we will find most of local cattle in the eastern province and the southern towards eastern southern area, and in the middle of the country we'll find European and their crosses, mostly find in these areas. So this is the distribution among 9 provinces. This is what I said. In some provinces, the dominating cattle type is local in northern province as well as eastern province. When it comes to central province, there are least number of local cattle. Again, this is from the statistical bulletin of Department of Animal Production and Health. I extracted data. Okay, so the exotic genotypes we have, obviously Friesian, Jersey Ayrshire for milk production, because we are for dairy cattle production in the country, because we are not talking about beef production here. So therefore, dairy dominating genotypes are there, and the locals are remain as nondescript. Though we call them nondescript, there are some attempts to describe these cattle. So I'm now from this point onwards, for 2-3 slides, I'm talking about nondescript or the native cattle genetic resources, because this is treasure that we have in our country, because we still think that it is an untapped resource when we talk about genetic resources. So the local cattle in southern and central parts of the country are towards brown color, brownish shades. When it comes to eastern area, it is white cattle, very famous white cattle. And there are some studies going on to evaluate the functional properties of milk of this cattle. So they are now looking at the beta-casein gene and the importance of that in their milk. Okay, so these are the two basic or dominant cattle types that we have, the native cattle line. But there are some geographically isolated sort of limited cattle types, and also the breeding taking place in these cattle in limited areas. So the Tavalam cattle is one of those categories of animals which we are now looking at, trying to evaluate their the genotypes and the genetic makeup. And there's another type of cattle, inherently small cattle. You can see that this is a grown-up man. The cattle is up to the hip of, not even up to the hip of the man. Okay, so this cattle not just we found. This is a picture of 1860. More than a century ago. See the cattle that they remain in this size. So there's some genetic the inheriting pattern going on. So therefore, we try to look at these indigenous cattle populations in different instances. So this is one of the initial study, looked at different geographically distributed native populations, and we found that they are very well separating from other zebu population in some parts of the world. And very recently, one of the colleagues in the Ovulation University, we looked at the Tavalam cattle, and we are still continuing with the genotyping attempts. So this is a very recent publication where we looked at the Tavalam cattle, and these are Tamankadu cattle and some other native populations of cattle, where the Tavalam cattle was having a very distinct position in the phylogenetic analysis. And this is the attempt we took to look at this the inherently small cattle. It was started very recently. The work is still going on, and it is going to be published in the World Congress in June. Okay, so let's move on to buffalo genetic resources. So we have very many number of improved type, and we have very few local. 12% of the population is represented by local type of buffaloes. So exotic breeds, the genetic resources that we have coming from Mura, Niliravi, and Suti, and their crosses. And actually, these pictures are about native type of buffaloes, indigenous buffaloes, which is a unique category. And we believe that they are descending from the wild buffaloes in ancient Sri Lanka, 15th century before Christ. There were some readings about these buffaloes. And why we talk about these Lankan buffaloes as a unique genetic resource, because they are phenotypically swamp type and genetically river type. And also they represent ancestors of present-day river buffaloes. So therefore, it is phenotypically very distinct population. So therefore, there were many studies went into that, went into looking at their the genetic makeup. But unfortunately, we have no any conservation program for local buffaloes. Other than these buffaloes were preserved in certain localities because their milk is mainly going for curd production, which is a cottage industry in southern part of the country. Other than that, there are no many programs going on to look at the resource of buffalo genetic resources in the country. This was an attempt in 1997, and where the Sri Lankan different populations of buffaloes compared with the other buffalo genetic resources in the Asian region. And other than that, there were few phenotypic characterization studies went into that, went into look at buffaloes, but no many studies done. So let's move on to goat and sheep animal genetic resources. The reason why I talked them together in the map, the Department of Animal Production and Health categorized them as small ruminants. So therefore, goat and sheep were taken into consideration together, right? But the composition wise, we have very different composition between goats and sheep. If you look at the goats, we have 0.36 million goats. Out of that, 60% are crossbred, Jamunapari crossbreds mainly, and 20% the improved the exotic genotypes, and we have about 20% local goats. And the goats are the improved goats are mainly find the Saanen mainly found in this middle part of the country. And when it comes to sheep are mainly found in Jaffna area. So that's why 90% of the sheep population, it is 0.01 million, very small number, and 90% of them are the indigenous type of sheep. We call them Jaffna local sheep. Only according to very recent calculations, it was about 900 something other sheep breeds that we found in rest of the country, rest of the parts of the country. So 90% of sheep are represented by Jaffna local native sheep. Okay. So let me take you through some pictures of goat animal genetic resources. So this is a lost pressure that we had Kotukachia. So no more Kotukachia specimens you can find, even though that there are few claimed to be, but they are not real Kotukachia specimens. And we tried to develop Sri Lankan boa category, but it was again not successful attempt. And we have this nondescript type of goats spread all over the areas of eastern and southern parts. Okay, so this unique native type of goats are very good genetic resource. So they are mainly for meat type, and they have very good record of disease resistance. And again, Ms. Kurukulu Surya is in her MPhil research, she was looking at the genetic resistance of the Jamunapari process and the native or local goats, and we found a very fascinating results in her research outcome. Okay, so we try to look at the phylogenetic, the structure of the phylogeny of the local goats with the other breeds in the rest of the world, especially in Southeast Asia of the Asian region. So it was published very recently, and there were two separate studies looking at the comparison of the Sri Lankan goats, the position in rest of the world, and also we looked at several populations of Sri Lankan goats and their genetic diversity. Okay, so coming to sheep, animal genetic resources. So I talk about Jaffna local sheep represented by 90% of the sheep population. And this is how they look like as of today. And there, we found that there was a very high level of inbreeding happening, and you can see that their ears are very, very small. That is an indication of inbreeding, we predicted. And they are preserved in a very unique farming system. They are only for manure. So because of this, the number, generating the number of individuals is something that farmers are looking at. We try to look at their the phylogeny, the composition as well, with the comparison of South Indian sheep breeds under international grant. And this is what we found. It was published very recently. You will find that Jaffna local sheep is represented by red color, and you will find that it is very nicely blended with the South Indian sheep breeds. And in this maximum likelihood analysis also, the main cluster is represented in very many places by the randomly by the Jaffna local sheep. So that indicates that the genetic resources are resource that we have in sheep, genetic resource that we are having are related to the South Indian sheep breeds. That justify the location as well, because they are limited to Jaffna Peninsula, and there are a lot of social connections, cultural connections between the two the entities, Jaffna and the South. Okay, so in genetic resources that we do not have very much here. Again, 0.09 million pigs we have. Out of that, 75% represented by improved breeds, the three breeds, large white land breeds and Europe. And we have about 25% representation from the indigenous pigs. The pigs, indigenous pigs like this, they are small, stocky, and produce very nice and flavor, the meat with nice flavor. Okay, and this is some genetic analysis we did way back by Miss Subalini, who unfortunately left us, so she is no more there. And so you can see that the improved breeds of pigs are very genetically distant from the wild pigs and wild boar we have. And we believe that village pigs are coming from or descending from wild boar. There are very close genetic relationship between these two, the native varieties. Okay, and we had a chance of comparing our pig genetic resources with those of Nepal and Bhutan in a small study. And there also we found that Sri Lankan pigs are clustering away from the Bhutanese and Nepal native pigs. Okay, so poultry genetic resource. So this is the vast area that again untouched, and we are still to sort of exploit the genetic resource that we have in this category. So here we are talking about mainly the chicken genetic resource, where we have a big diversity. Okay, so if you go by numbers, the large numbers of chicken genetic resources are represented by the exotic strains and lines. So more than 85%, the number-wise, more than 85% are represented by these improved varieties. And so I said that chicken is dominating. Other than that, we have small numbers of ducks, turkeys, guinea fowl, and quails. And also the indigenous chicken types that we have representing about 15% 15% of the total chicken genetic resources in the country. Okay, so that 15% is very rich in diversity. So we have very many different types of chicken in our village plot. Okay, so there were attempts in describing these different types of chicken, indigenous chicken like neck and neck, long leg, frizzle, crested, fighter, and some looked at the distribution and the distribution pattern. But we could see is that all over the world we have all over the country we have a mixed genotype, and there was no sort of a separation of genetic differentiation that we could find in different geographical areas, in different types of chicken, even though we phenotypically describe this as neck and neck, crested, fighting, and long-legged, but genetically they are almost similar. Okay, so this graph, the map will describe you what I said, and it is based on a study that we did by looking at different places of the country and sampling them in from different places. So we found that it is a mixture of different genotypes in all the places. Okay, what we found in some of our studies is that even though that Ceylon jungle fowl is considered as one of the ancestral species that contributed present-day chicken population of the world, our native chicken has no relationship or has a very limited relationship with Ceylon jungle fowl. So this is a phylogenetic analysis that we did with the Ceylon jungle fowl, and you will find that our native chicken are clustering very close with the red jungle fowl, which is another ancestral breed, and also the gray jungle fowl. So there's no connection to the Ceylon jungle fowl. Okay, so these are the main species, main animal genetic resources that we have. Other than these species, we have very fascinating genetic resources in our country which are untouched. I want to talk about Delphoni. I'm just showing you pictures because there are no very many studies done on them. Only the phenotypic description is there. And the manna donkey. Again, even the phenotypic description is also not available for manna donkey. And pigeon. Okay, so it is a big genetic resource. Mind you that all these pictures are from our country. So we have very many type of pigeon. In the western coast, there's a big sort of sport club or the society is established, and they are doing the breeding and exchanging these birds for different purposes. Some of them are trained for racing, some of them are trained for performing, and some are for fancy ornamental purpose. Okay, so let me wind up by just touching on the wild relatives of our the main genetic the domestic animals that we have in our country. Some of these the domestic species have originated from native ancestors, so therefore we have their wild relatives, and also we have evidence for evolutionary history, and some other have been introduced long time back, though no wild relatives. It is blends of different genotypes. So if we take cattle, there's no ancestor exist, and we have some sort of belief and also some evidence to and one of the extinct relative of cattle, that is Gavara cattle, and this is droving that somebody made, I think Dr. Bagnigel made by getting the fossils. Okay, so this is again a lost treasure, the Cape cattle. I can very well remember when I was a child, my grandparents are talking about Cape cattle. So again, we do not have the Cape cattle anymore. It is extinct and it is included in the extinct list of FAO. So therefore, evolutionary path of the indigenous cattle, present day indigenous cattle is not known. Buffaloes, we do not have the ancestors. The wild relative is called Kulumima is there, but there's no sort of evidence to show that the ancestor, the descending of present day buffalo from this wild ancestor. So the pigs, we do have the ancestors, that is Scrofa affinis, the wild boar, and there's a close genetic relationship with the indigenous pigs. And the jungle fowl, as I said earlier, there's no relationship between the indigenous chicken and wild relative. And again, this is untouched and unknown wild genetic resource, the Lankan wild rabbit. No any evaluation went into this genetic resource as well. Okay, so therefore, we have animal genetic resource, vast array of animal genetic resource, was diversity, with what was diversity, and we consider it as a big resource, but we have a lot of unknowns, breed level trends that we do not know, because we are tracking only the categories, the exotic and local, or else Indian crosses and the European crosses. So the breed level trends are not tracked, or we do not have any idea of it. And population trends of native animals, we do not know. We have very limited documentation. Risk status, we have not yet studied. Conservation needs, we have to identify, and characterization for important genes still that we have to go along with. So in my concluding remarks, we have highly diverse animal genetic resources in our country, and there should be a national level focus on studying dynamics of animal genetic resources, and individual population trends are to be monitored, and unique variants in native populations are to be assessed. So that is my concluding remarks. Thank you very much for your patience listening. Thank you.
Thank you very much, madam. And thank you very much for sharing wonderful presentations with us. And there's a lot of information regarding the present status and of the animal genetic resources, particularly in Sri Lanka. So I would like to announce the audience, since Professor Silva not attending the panel discussions, we are planning the later. So, but you can raise your the questions, and feel free to ask any questions from the Professor Silva by unmuting yourselves, or you can send your the questions through the chat box. So this is the time for your questions.
Looks like I made myself clear.
Yes, ma'am.
Madam, I'm Sudhini.
Yes, Dr. Sudhini.
Madam, now it's very clear that we have a good genetic potential, but however, still we are kind of far behind from the other countries in terms of production. So does it, is it a problem related to our genetic potential, or does it belongs to any other management aspect?
Thank you very much, Sudhini. It's a very pertinent question. I like your question. Yes, we do have a good genetic resource base. The thing is that the phenotype is the blend of genotype and the environment. So when we talk about cattle, I think you were referring to cattle because we are struggling with our milk production. So in my opinion, the problem is with the nutrition and the input supply. And even though that we have a potential, the potential cannot be tapped if you do not provide them with a conducive environment. So in the case of cattle, if I take cattle as an example, we are struggling because of the input supply and the nutrition supply. So we are not sort of poor in genetic resources, but the thing is that we are poor in some of the management aspects and the strategies. That is my answer to your question.
Madam?
Yes.
Madam, I have one question.
Go ahead. Yes.
Since we have talked about a lot of genetic resources, and still we have to go beyond the current status, right, madam? So in that case, particularly the conservation side, and then also the characterization of specific genes that we have to go for, like disease resistance, on how we can, if we improve the nutrition of the particular livestock. And then we have to check whether these nutritions and the particular genotypes have the relationship. For example, like the quantitative traits or the relationship between the nutrition and the genetics. In that case, how we can go and what kind of potentials we have in that perspective.
Yes, very good. Thank you very much for your question. Actually, we have to have a planned breeding programs, and especially with the selection. Okay, so even though the genetic base is there, we cannot explore the potential of it if we do not allow it to sort of surface. So the selection is the way to surface the hidden genetic potentials. So we have to have a very clear sort of breeding program. If you take in our the farms, say the government farms or the private sector farms, what we do not see is a good breeding selection and breeding program. So we have to have a sort of system in evaluating the genetic potential of different categories of animals, not only production potential, but as you said very correctly, the disease resistance and all type of these important genetic potentials that we have to have a program for evaluation. So simply the breeding value calculation. So if we take the main industry like milk industry, so we do not have the breeding population genotype and the breeding values calculated. There are attempts going on in Department of Animal Production and Health, but we have to have proactive in this area to do a proper selection, and then we can identify these genotypes where they are more suitable in getting the best out of it.
Thank you very much.
Okay, my pleasure.
Hello. Can you hear? I'm
Yes.
Sunil Gamage.
Yes, Sunil Gamage.
Yes, I have a small observation and a correct this sort of a question also. Thank you very much for a very good presentation, Prof. And this is during the last 20 years. You said that you mentioned that the department will have or the government will have to conserve our indigenous animals, but in the past this was done by the keepers. It was not done by any government. But then we make it difficult for them by circulars like 2005, 2000/5. We lost about 65% of the indigenous animals because most of these animals were free grazed, and the herd free grazing was controlled by this conservation. I have the data, and I'm sure you may also have the data. We lost around 65% of indigenous animals. Now, fortunately, they have removed that circular now. So free grazing is allowed in pasture lands again. And why such decisions are taken by governments based on looking at one area like forest conservation, and it has not only it has really affected the farming population in this country. So my question is, how are we going to get the herders back into business after this?
Yeah.
Thank you.
Thank you, Sunil. Sunil is a very good friend of mine for a long time. Sunil, you may be able to answer the question more than me, better than me. But if I try to answer your question, yes, the haphazard decisions, I mean, cause a lot of sort of situations and putting the whole industry into trouble time to time. So as you said very correctly, that the free grazing, the prohibition of free grazing hampered the livelihood of many farmers in our rural areas. So that affect the genetic resource and degradation of genetic resource and erosion of genetic resources a very big manner, because farmers tend to now keep the only improved cattle in small numbers, thinking about their difficulty in finding feed. So as you very correctly said, until the time that we get the decision makers onto one table, and also all these, the ministries are identified not in a scientific way at all in this country. Until we get such a time, we had to wait. I don't know, Sunil, it is in your lifetime or my lifetime that will happen. But in one fine day, if we get all the decision makers doing their decision making in a identifying the interrelationship of the whole habitat of preserving animal genetic resources, we have to sort of fight for this sort of thing. So unfortunately, I have no very clear answer to your question, but I am having the same position as you have. Yes, we are facing a big trouble with this haphazard decision-making process.
Thank you, Prof.
Thank you, Sunil.
Thank you very much, madam. And I would like to take time to thank you for joining and sharing your the thoughts and the recommendations and the knowledge you have experienced through your career. So we are very honored to having you in these sessions, and thank you very much, madam.
Thank you very much. My pleasure.
Without taking much time, so let me introduce the second speakers of the forum today. The second speaker of the forum is Dr. P.G. Senivaratna, the Director of Animal Breeding in the Department of Animal Productions and Health, Peradeniya. Dr. Senivaratna obtained his bachelor degree in veterinary science from University of Peradeniya and his MPhil in the animal reproductions from University of Pedigree. Dr. Senivaratna joined to the Department of Animal Productions and Health in 1993 as a veterinary surgeon and served in different institutes. Dr. Senivaratna served in Central Artificial Insemination Station in Kundasala And also he served in livestock breeding project in Polonnaruwa as a project coordinator. As well as Dr. Senivaratna worked as animal breeding division as an assistant director in dairy development and deputy director in animal breeding divisions. Not only that, Dr. Senivaratna has been participated and received international trainings on swine buffalo production, crossbreeding and sire evaluation, suitable cattle breeding improvement, and the technical management of artificial insemination and molecular genetic characteristics of domestic animals. So I would like to kindly invite Dr. P.G. Senivaratna to address the forums on the topic of the pros and cons in utilizing animal genetic resources in livestock production. Yes, sir, over to you.
സംശയം എന്നാണ് Okay.
Yes, sir, we can hear.
Okay, can you see the screen now?
Yes, sir, we can see.
Okay, thank you. And thank you very much for introducing me and giving me the opportunity to discuss with you about pros and cons in utilizing animal genetic resources in livestock production. So animal genetic resources of all animal species and bird breeds of economic and scientific importance to mankind in terms of food and agriculture products is the definition for animal genetic resources. So utilizing animal genetic resources is the process of discovering and exploiting the beneficial features of genetic resources in order to develop livestock products. So in any country, the utilizing animal genetic resource will require systematic and coordinated breeding programs based on thoroughly evaluated national breeding policies. In Sri Lanka also, we utilize animal genetic resources based on a policy formulated by a committee known as National Animal Breeding Committee. So as for the recommendation given by the committee, animal breeding is being done to produce suitable dairy type animals that could be adapted to various agroclimatic zones. In Sri Lanka, there are 7 agroclimatic zones, namely upcountry, wet and intermediate zones, mid-country wet and intermediate zones, low-country wet, intermediate and dry zones. So first we'll look at cattle breeding policy. The recommended for first we'll see what's the recommendation given for upcountry wet and intermediate zones. The breeding objective is to produce dairy type animal, and policy is grading up using temperate breeds. The grading up of existing cattle using temperate breeds, and breeding strategy is defined separate for different management systems. The management system existing in this area intensive and semi-intensive, and breeding policies continuous upgrading using European breeds. The recommended breeds are Friesian, Ayrshire, and Jersey. Next, we look at mid-country wet and intermediate zones. The breeding objective is to produce dairy type animal. Breeding policy is to grading up of existing cattle using temperate breeds. Breeding strategies defined separately for different management systems. The existing management systems are intensive, semi-intensive, and extensive. Continuous upgrading using European breeds is recommended for intensive management systems. The recommended breeds are Friesian, Ayrshire, and Jersey. Continuous upgrading using Jersey or grading up to 75% blood level using Friesian or Ayrshire is the recommendations given for semi-intensive management systems. Grading up of existing cattle using temperate into Zebu, crossbreed bulls, or continuous grading up using Sahiwal is the recommendations given for extensive management systems. And the breeding policy recommended for low country wet, intermediate, and dry zones. The objective is to produce dairy type animals. Breeding policy is to grade up using temperate breeds. Breeding strategies defined separately for different management systems. Here also we find intensive, semi-intensive, and extensive management systems, but continuous upgrading using Jersey or using European breeds is recommended for extremely high level of intensive management systems. So here you should have environment control systems to manage this type of European breeds. Grading up using temperate breed to produce 50% European level is recommended for semi-intensive management systems in this area. Continuous grading up using Sahiwal is recommended for extensive farming systems. So that's about the cattle breeding policy. And now we'll talk about buffalo breeding. The recommendations are given for the entire country. Breeding objective is to produce high quality dairy or dairy dominant dual type buffalo. Here dual type means the draft purpose buffaloes. Breeding policy is grading up of using herds, using existing herds using exotic breeds of buffaloes. Breeding strategy is common for all of the farming systems in the country. The management systems existing in this area are intensive, semi-intensive, and extensive systems. And the recommended breeding policy for all management systems is grading up of existing buffalo using Mura or Niliravi breeds. Now we'll talk about goat breeding policy. Here also the recommendations given for the whole country. Breeding objective is to produce dairy type animal in upcountry wet zone. And to produce dairy type or dairy dominant dual purpose animals in upcountry intermediate, mid-country, and low country zones. Breeding policy is grading up of existing cattle using exotic breeds. Breeding strategy common for all farming systems in upcountry wet and intermediate zones. and breeding strategy depend on the farming systems in mid-country and low-country zones. So breeding policy is continuous grading up using San and breed is for the upcountry wet zone and intensive and semi-intensive farming systems. Continuous grading up using San and Jamnapari breed is recommended for upcountry, intermediate zone, intensive and semi-intensive farming systems. Continuous grading up using San and Jamnapari breed is recommended for mid-country and low-country intensive farming systems. And continuous grading up using Jamnapari breed is recommended for mid-country and low-country semi-intensive and extensive farming systems. Right, now we'll talk about pig breeding policy. The recommendations have given for the whole country. Breeding objective is to produce fast-growing commercial pigs. The breeding policy is to maintain existing exotic breeds. The existing exotic breeds are landrace, large white, and duroc. To maintain existing exotic breeds of pigs and to produce suitable commercial pigs is the breeding policy. Breeding strategy is crossbreeding, two-way or three-way crossbreeding for commercial production. The recommended management system is for the whole country's intensive system to produce two-way or three-way pigs using large white and landrace and duroc breeds. The two-way pigs would be the crosses between landrace and large white or vice versa. Three-way crosses being produced by crossing the landrace large white crosses with duroc as terminal sire. So that is the recommendation for pig breeding. Now we'll talk about the topic given to me about pros and cons in utilizing animal genetic resources. The utilizing animal genetic resource in livestock production is vital for food security and rural development. It provides people with a variety of products including meat, milk, egg, fiber, etc. Animal genetic resources are used in animal breeding to achieve the task given below: increase the quality and quantity of livestock production, improve the growth rate of farm animals, have strong resistance to various diseases, to develop longer productive life, lead higher acceptable reproductive life. So we use crossbreeding to achieve these tasks, and crossbreeding used to develop more superior breeds and also to improve the quality of local breeds. The hybrid thus obtained, we have more commercial value. We talk about the use of exotic breeds of cattle in crossbreeding programs. So this crossbreed, the exotic breeds improve the quality and productivity under favorable conditions, but under tropical conditions, they are relatively low fertile because of their poor adaptability. And they have high mortality rate, low feed conversion rate, less resistance to the diseases, and high maintenance requirements. Therefore, although dairy cows are genetically improved, their productivity decreases if they are not properly managed. Hence, cost of production will be increased. If we talk about the use of indigenous livestock breeds, though they have many favorable characteristics like high fertility, low mortality rates, disease resistance, their production potential is low. So the total income of the farmers will be low. So we are doing animal selection. People deliberately practice selective breeding to develop livestock more suited to their needs that allows producers to select and breed parents with desired traits to produce offspring with more desirable characteristics. That means genetically superior to the parents. The successful results of selective breeding throughout the years can be seen in livestock production today. For an example, in the livestock industry today, cattle are also evidence of successful selective breeding. Dairy and beef cattle are both highly efficient animals that produce more milk and meat in the years past. So if you look at this graph, you will find that during the last 10 years period, the cattle population has slowly declined. At the same time, the milk production has slowly increased. We assume that this may be due to the genetic improvement of the national herd. In addition to that, the genetic improvement can be achieved by selecting genetically superior animals to be the parents of next generation. That will lead to reduced genetic variation, mainly through the widespread use of small number of superior animals. And another problem is inbreeding, which can cause genetic variation within the breed decrease. Another problem is with crossbreeding, the genetically transmitted diseases can appear in the progeny. For example, blood, Shetlonemia, DAMS, factor 11 deficiency syndrome. These are genetically transmitted diseases by autosomal recessive genes. If the parents are heterogeneous, these diseases will not appear in the parents, but which could be appear in the progeny. So this is another problem with genetic improvement programs. The other thing, as Professor Pradeepa mentioned, if you do not properly manage, some of the important breeds will be disappeared. For instance, the Hatton cow or the Cape cattle, as Professor Pradeepa mentioned, now already these breeds are extinct. So now I explain the animal breeding policy already. So in Sri Lanka, we use animal genetic resource for improvement of the national herd based on the animal breeding policy I already explained to you. So we disseminate the recommended genetic resources through our national AI program and natural breeding program. And we have 3 other supportive programs: Pedigree Performance and Recording Scheme, implementation of heifer calf rearing program at provincial level, and promote feed resource development and utilization. So I'll just give you a brief introduction about our national AI program, artificial insemination program. So we facilitate this program at national level by providing required inputs like Siemens and other consumable items. And we have 2 AI centers for Siemens processing. One is at Kundasale and the other one is at Polonnaruwa. And also we do semen importation time to time for the introduction of new genes to the national herd. And we process semen from Jersey, Friesian, and crossbreds. Also, we have introduced Girolanda crossbreeds for the national AI program recently. And as I mentioned earlier, we import semen time to time to introduce new genes to the national herd. This is a measure to prevent, one of the measures to prevent inbreeding in the national herd. And the other program is natural breeding program for genetic improvement. We implement this natural breeding program in dry zone area in large herds. Since the AI service cannot be accessible in this area because in large herds there's no possibility of heat detection. So that we introduce good quality bulls for natural service into these herds. So now I will talk about the other supportive programs as I mentioned earlier. And first is Pedigree Performance and Recording Schemes. In short, we call it as PPRS. Here, this picture depicts the summary of that program. I'll give you a brief introduction about that. From the base population, we have selected some cattle or some dairy animals based on certain criteria, and we do milk recording of these animals. And out of these animals, we select the best animals and use imported semen and produce bull cows. And the top bulls will go for the National AI Center for semen production, and the rest of the bulls we use for natural service in the base population. So that is a summary of the PPRS program. So we are implementing this program by our division. These are some bulls selected through the PPRS program as semen donors. They are all crossbred animals. And we have another program called heifer calf rearing program. This is we implemented to salvage AI-born heifer cows through improvement of their nutritional status. And almost all AI-born heifers we are registering and provide some incentives to achieve optimum reproductive parameters. So those animals who achieve the recommended reproductive parameters will receive the incentives. And the other project is promote feed resource development and utilization. We introduce new varieties of pasture and fodder to provide good quality feeds to the improved animals. And we have goat breeding program as well as swine breeding programs. We maintain 2 nucleus herds. One is Boyer breed and the other one Jamnapari, and we provide breeding materials to the national herd for herd improvement purpose. The swine breeding program is mainly handled by the Western Provincial Department of Animal Production and Health because the most of the pig population is scattered in that area. And as central government, we are facilitating this program and it implemented by the Western Provincial Department of Animal Production and Health. I think that's the end of my presentation. Thank you very much for being with me and bearing with us. Thank you very much.
Thank you very much, sir. So since we have another panel discussion at the end of the session, so we would like to have the discussions later, and then I would like to go for the next presentations of the today forum. Let me introduce the next distinguished speakers of the today our forum. Professor Suranga Kodituaku. Professor Suranga Kodituaku graduated from Animal Science Department, University of Peradeniya. Professor Kodituaku graduated from Faculty of Agriculture, University of Peradeniya, Sri Lanka in year 2002. animal science. He receives further training in animal reproductive biology and molecular biology from Obihiro University of Agriculture and Veterinary Medicine, Japan. Later, Professor Suranga completed his PhD in reproductive biology at LKS Faculty of Medicine, the University of Hong Kong, Hong Kong. So Professor Kodituaku is actively engaging in research area on animals and the human reproductive biology, molecular and cellular biology. Present, he is attached to the Institute of Veterinary Medicines and Animal Science, EMU Estonia, as a senior research fellow under the European Union ERA chair in competitive medicines. I would like to warmly welcome Professor Suranga Kodituaku. He will be presenting on the present and future status of animal reproductions and biotechnology in Sri Lanka. Sir, over to you.
Thank you very much. May I have the rights of sharing my files, please? Yeah.
Yes.
Can you hear me well, and can you see my slides in the presentation mode?
Yes.
Yeah, okay, thank you very much. Thank you very much for your kind introduction and also inviting me to be part of this forum today. So today I'm just going to share a little bit of thoughts on present situation and in terms of reproductive management of the livestock in Sri Lanka. And also specifically, since we are mostly these days concerning about the dairy production and the cattle production. So because of that reason, I'm going to be more focused on that. Plus, I'm going to share some of thoughts on how the biotechnological intervention can be done, to improve the present status of the reproductive management in the country. So if you look at all around the world, actually, it's not something that is unique to our country, but everywhere else where the animal or livestock production is being done, we have issue of reduce of fertility in farm animals. In some species, actually, for example, like cattle and especially like horses, especially in this European Union area, where we have a serious issue of reduction of fertility. So year by year, actually, we see reducing trend. That's not very healthy in terms of the sustainability of the industries. But of course, we need to deal with this issue. And obviously, as a country, Sri Lanka too has the same issue, but we are facing it in a bit severe manner than the other countries around the world because of some other associated unique issues associated with the Sri Lankan situation as well. Let me begin something with the cattle reproductive management, and all over the world, actually, we are focusing this 100-day program in managing dairy cows. In terms of reproduction efficiency, we are trying to get a cow pregnant at least 100 days after the calving. So that's what we call 100-day program of dairy cows. Actually, this is not only dairy, but also for beef cattle. The same program exists elsewhere in the world. So in this program, what we actually focusing is we try to, phase out these 100 days into different categories or different phases. And then we are trying to manage these cattle differently in each of these phases in order to make sure that we are going to have a pregnancy at least by the 100th day from the calving. So it involves actually dry cow program. Of course, you may all know by now, actually, maybe some of you may not know. Dry cow program is actually meant to be to flush the animals and to make the animals at the end period of the pregnancy, just to make sure that they are going to have a good calving and afterwards good milk production. But obviously, dry cow program is also linked with the reproduction management as well. I'll tell you why and how in some of the slides coming up in my presentation. And then, of course, there's a period called fresh cow program. Soon after the parturition or the calving, the animal is undergoing a transition status where the animals need to be taken care differently than the dry cow period. And then afterwards, with the time, as the lactation going on, then the animal can be entered into the breeding phase. We are trying to get the animal pregnant again, right? To make sure that the production will be continued in the next cycle as well. So this is what actually right now the focus in the dairy industry as well as meat cattle industry. But even though we have this program, still the industry is actually engulfed or threatened with some of the issues, inherent issues. This has been there. These issues have been there for a long time, and people have been struggling to overcome some of them. But still, we have some solutions. But still, I would say we are not 100%, competent in solving some of these issues. So if you look at the global picture, anti-stress, especially postpartum anti-stress, where animals are not showing heat signs after the parturition, is one of the key concerns. Of course, an ovulation where animals are not ovulating. Plus, we have serious issues of success rates in artificial insemination and embryo transfers, which will actually lead into the low conception rates of the animals. And we see repeat breeding because of the same issues. And of course, especially in some part of the world, including our country, we have some difficulties in detecting pregnancy as early as possible. Because why it is important to detect pregnancy early as possible is that if you detect animal is not pregnant, then obviously you can just put the animals back into a breeding program without losing open for longer period of time. Ultimately, all these issues will lead into something called extended calving intervals, where in Sri Lanka, I will talk to you in the next slide, where we have a serious issue, right? And of course, if you look at the economic terms, 1% decrease of this pregnancy rate of a herd will severely impact on the economies of the farm. So if you just, but unfortunately, we don't have such calculation done in Sri Lankan terms, but obviously, if you can just look up the literature all over the world, you can see huge impact on even 1% decrease of fertility rate in cattle. So if you look at the Sri Lankan scenario, the longer calving interval is the main issue if you talk about the reproductive management. Longer calving interval, meaning we would like to have about 1.1, 1.2, at least 1.3 years of a calving interval from one calving to another. But obviously what we see is the picture is not that rosy, because all over the country, if you go and talk to the farmers, if you go and talk to even the large scale farms, even private or public sector, you can see most of them are having the serious issue of longer calving interval, where sometimes it goes up to even 2-3 years, but unfortunately you cannot cull those animals and they have to keep them and trying to get them pregnant as well. So if you look at the reasons for these longer calving intervals, actually you can list out many. It is not exhaustive list, but just I listed out the key issues, contributing to the longer calving interval. One, like I mentioned in the previous case, like in many parts of the world, we do have the postpartum anti-stress issue. And we have a significant issue of negative energy balance in our cattle. I will explain in the next slide what is negative energy balance, because in good old days, we did not pay much attention on this negative energy in terms of reproduction management. But right now, all over the world in the dairy industry, in the cattle industry, now we are seriously focusing on negative energy correction in order to achieve good reproductive success. Of course, we do see the repeat breeding, AI failures. Last 5-6 years, we have been doing a large project in Sri Lanka and improving dairy production, where we look at in different angles of the dairy, like from reproduction aspects, hygiene, nutrition, welfare, all these aspects were there. But we found that alarmingly, even some farms, even well-maintained farms in the country, where they do sometimes 7-8 AIs per pregnancy, which is no way having, if you look at the economics of the farms. Of course, we do have issues of heat detection, especially in large herds. Partly, I will tell you later, because most of these things are coming up because we are not that, receptive for some of these technologies, maybe because of the financial reason or otherwise. We are not using, correct technologies so that we are into this kind of issues. Of course, AI failures are there. And we do have some difficulties in doing AI timely sometimes because of logistic reasons, reasons beyond sometimes control of the farmer. And of course, we don't have a very active or good embryo transfer program. The country, instead, we are importing animals to the country to improve their heard, but in my personal opinion, I think it should not be the way, but we need to have our own embryo transfer program in order to uplift or upgrade our own animals with some exotic blood. And we have issues of, availability of sex semen because farmers prefer to have female calves. So that I think there are certain level of availability, but I would say it's not that accessible and freely available to everyone. And it's costly actually. There are certain subsidy programs running in, but still that is not enough to cater the demand. And of course, the reproductive diseases like endometritis is having a direct link to the reproduction efficiency of the cattle. So we do not run a very sound program to identify endometritis-like condition, especially subclinical conditions. Of course, clinical condition at the clinical level, when you have signs and symptoms, of course, there are treatment options available, but we don't have any screening programs like in European Union and any other countries. They just routinely do this, screening for the cyto, like early subclinical level endometritis diagnosis, but we don't have such programs running in our country. Of course, we have seen a lot of an ovulation problem issues plus cystic ovaries in our cattle, but that is actually partly linked with the negative energy balance, where the impact of negative energy balance sustained for a certain period of time, resulting in ovulation actually. The other issue that we have is monitoring the semen quality at the field level. I have some data, some of my senior colleagues, Professor Missaka did one nice study sometimes back. And we found that even at some places in the country where the mortality of the semen is actually 0% at the time of AI, because of various reasons, lack of storage condition or proper storage, sometimes improper handling of the semen stores. Those conditions, the things will contribute to this kind of problems at the field level. But so we need something on ground to evaluate this and monitor this as well. Mastitis is a serious issue in the country. I will share some data, very recent data that we have. And in good old days, again, like in endometritis and negative energy balance, we did not pay much attention on mastitis in terms of reproductive management. But right now, everywhere in the world, mastitis is a key thing that we focus when we want to have a good reproductive management program, because there's a direct link with the mastitis plus reproductive outcomes, especially the conception rates, because of the biologies involved in the disease etiology. And of course, I think Professor Pradeep also mentioned, and the deputy director from the DA also mentioned, we have some issues of availability of having capacity of producing high quality semen. Sometimes we do have some in the country, but I think it's not enough for the time being. So what we can do, yes. If you just focus on these three phases of this 100-day program, if you just address some of these issues that we have seen, which I have listed in the previous slide, there are some remedies we can do in order to achieve this 100-day program. So we have some options of addressing the negative energy. We can do very simple things to overcome the negative energy, and that will impact the transition cows, and that will improve the cow's fertility. They will come into heat very quickly, and also they will have a very high success rate even in subsequent artificial insemination. And obviously, without waiting for, looking for estrus signs and symptoms, we can obviously go for the ovulation synchronization protocol. Mind you, this is ovulation synchronization, not the estrus synchronization, because in our country, mostly what we see at the ground level is using PG-based estrus synchronization protocol due to various reasons like unavailability of drugs, accessibility for the drugs and veterinary services hindering having a very efficient ovulation synchronization programs. But obviously, we can overcome this by having such programs. And thirdly, the pregnancy detection. In most of the time in our country, we are depending on the manual pregnancy diagnosis using rectal palpation, but it is not that effective when you want to put the animals back, especially non-pregnant animal, back into the breeding, where you cannot let the animals wait for 60 days or 45 days after AI. If you want to have a very efficient reproductive management program, you need to detect whether they are pregnant or not as early as possible. For example, if I can do it by day 20 or day 28 after AI, that would be the ideal situation where non-pregnant cows can be put back into the synchronization programs. Since I wanted to stress a bit on negative energy, actually now there's a huge interest and demand all over the world on this nutritional reproduction in cattle management, where nutrition is considered part of actual reproductive management. During the peri-parturition period, just before a couple of weeks prior to parturition and a couple of weeks after the parturition up to the peak milk production, the animal is undergoing a huge energy balance issue. Even if you look at the animal where the body condition score is perfect, like maybe 4, 3.5, right? But inside the animal, in physiological basis, if you look at the animal is going this negative energy issue crisis. So it's not something that you can look at from the surface or from the outside and judge. It has to be done with some empirical or testing evidence, with some need to do some testing and then need to identify. If you manage this period with supplementing the energy properly, obviously you can just get the animal pregnant within before 100 days of the calving. We have the negative energy balance issue in our country. Yes, I think you can be proud of one of your colleagues from Usda did this work with our research program, where they have screened a lot of animals in the country, small scale, medium scale, large scales, and they did find, or we did find that there's the cows in the country are undergoing this negative energy balance, negative energy situation. So we need to address this, but there are solutions, and obviously we need to have certain platforms to identify. So blood chemistry is the most trusted method of identifying negative energy. Obviously, we need to use some kits, ELISA-based method. And I believe that at least we need to provide this service at the VIC levels in the country, even for a normal or individual farmer to have the access and get the animal tested soon after the parturition, at least, to get an idea. And obviously we have some certain avenues in the local market. We can develop these kits in our own kits, especially at the time where we are undergoing severe shortage of foreign currency, where we don't have the luxury of importing them. So, but these are not very, sophisticated kits. They are very basic kits actually. Even if pharmaceutical companies are interested, there are avenues of producing these kits at local level as well. And obviously we need to do validation and then use it afterward. After you decide or detecting this negative energy balance, then what you can do? There are solutions, obviously. Of course, you can supplement the energy. And again, our colleagues did this nice study sometimes back, and they found even if you supplemented with just 200 grams of, rumen bypass fat, obviously you can recover the animals. Even the farmer can get more income. Plus, we observe improved reproduction performances where we were able to, get back into the cycle very quickly, and then we were able to get them pregnant with 1 or 2 AIs without wasting or without keeping them open. Producing this bypass fat as well as bypass protein, rumen bypass protein, is one big avenue where we can use some of the biotechnological interventions, especially like microbial enzyme-based fermentation techniques can be very easily used, and there are tested, proven technologies even available that we can easily adapt to, so that if we can detect the issue in that. The second thing, actually, to have a better synchronization. That's what I mentioned in one of my previous slides. So we do have many programs going on, especially in the private sector farms. Plus, we did have some programs with these imported animals. Again, a company involved in, and so they tested some, and they introduced few of their own ovulation synchronization protocols for the country. But still, if you look at, there are no very strict recommendation or guidelines as such at the moment, because still we're not that great in using these in managing our. There are many programs where we even tested these three systems, these three protocols very recently. And when you look at, if in the normal farm level, these were the performances, if you just introduce them without proper guidance, you can see the success rates are even like just around 10% sometimes. So it's not great. Even you spend a lot of money, but still you are not getting anything out of it. But what we found was actually, this is not because of the fault of the program that we introduced, but of course, it's because of the nutrition actually. If you not manage the negative energy properly, and if you did not supplement them with enough adequate energy, so this would be the outcome, even though you have the the best possible protocols available on that. So that's what I said. Energy and the nutrition and the reproduction need to go hand in hand if you want to have an effective and efficient. And obviously, we also tested some other like supplemented progesterone, whether we can improve. But what we found was, of course, it's not really the protocol that matters. Actually, it's the how you manage or maintain the negative energy would be the key to producing crossbreed animals, even recently imported ones. So adding more money to the treatment will not help you if you do not manage them properly in nutritional wise. But if you just manage properly, plus if you perform the best protocol available, of course, you can, even in one cycle, you can go up to 40%. And in some instances, we were able to go up to 50% success rate in one cycle, one synchronization cycle, which is really great, even in par with some of the data that we have in European as well as in other countries. So the third one, actually, I mentioned that we have issue of, detecting pregnancy because we mostly depend on the manual PD. And this accurate detection is very important because if you just let the animal to be open, if you do not know whether the animal is not pregnant, that is the case. If it is pregnant, it's okay. But if the animal is not pregnant after doing an AI, if you don't know that animal is not pregnant, that is the issue. So you can see some of the economies, some of the calculation where how much loss you can incur if you have a single day of open. But obviously, again, like I mentioned, we do not have any calculation done in rupees and cents in the local condition. Obviously, we have to always refer side data if you want to get impact of this kind of single open day, so prolonged open days. Standard of detecting or diagnosing pregnancies using ultrasound scanning. Obviously, whenever I throw this word out in the Sri Lankan forum, oh, that's very expensive, you can't do it. It's too much for us. We're a poor country, but it is not actually. Now we have very nice handies to use that you can use. I will share some of them in the next slides, where you don't need to have big equipment like we have been using in our research, but of course you can do it in a very modest way with same accuracy if you want to do it. And obviously you have to rely on some biomarker, serum biomarker based kit-based detection, where even an individual farmer can use very easily. But obviously you need to give them training, or at least you have to have kind of trained personnel available. I will show some information later where still in Sri Lanka also now we are using two kits, Bioprint and IDEX1, for this very purpose, very successful. So there are many kits available based on serum biomarkers. People depend on different biotechnologies, different companies adopted, they have different detection platforms. These are not 100% effective. Sometimes there are certain pros and cons in all these kits available, but at least once we tested, obviously you can find out what is the best for our own conditions, and you can use them for the efficient use, especially like this IDEX1, the PAG9 based kit, which we have tested and also some other people have tested. We could easily use in our country, and it is available in the country right now with another kit called Bioprint. But there are certain limits in these kits where you cannot use it for cows within first 60 days with the presence of the PAG9 protein in the blood circulatory system from the previous pregnancy. But obviously in heifers, even within 28 days after AI, you can detect the pregnancy. After 60 days, obviously you can very nicely detect the pregnancy in the cows. We have done all our own work also identify new markers with the aim of actually developing new kits. And we found that even in our condition, ISG15 would have been much better than the PAG9. And actually, we are right now in the process of developing this our own kit, where even the farmer can use at his own home using the milk sample. So we have some issues of logistics and some of the technological mutations in the country right now, but we are hoping to develop. I mentioned that the health issues like mastitis and endometritis, again, key to, if you properly manage mastitis, of course, you can get the benefit into your reproductive management program. You can see this data from our own research group. Dr. Rasagadian Dasa led this study actually in almost all the cows, I would say, in Sri Lanka, and he found, you can see, we have a serious issue of subclinical mastitis. So with this mastitis, and if we have the negative energy, I don't want to tell you the efficiency that we can reach. So obviously there's an issue. We need to have a very, of course, testing is needed, screening is needed, plus the other issue comes with, even you start treating these animals, there's a huge issue of antibiotic resistance in these mastitis organisms in the country right now because of the improper use of the antibiotics. So we may need to look into very seriously and try to overcome. If we want to have a good reproductive efficiency now in our dairies, obviously this is one thing that we need to have more focus, not only the reproductive issues per se, but also some of the health issues, including the mastitis. So I have listed out some of the interventions we can adopt to improve the country. So this is not, again, an exhaustive list, just key intervention which we can do. Of course, we need to produce our own embryos. That is one key thing if we want to have a good upgrading program. Not importing animals, obviously, is the solution, but of course we need to produce our own. Of course, if we can do the sexing of embryos using biotechnological means, that would be the greatest thing. But again, we have to go extra mile. We have the know-how, we have the expertise, we have everything. Only thing is big time investment from the government side, at least, to have this kind of program. I know there are some programs running in the faculty to start some good embryo transfer program, but still they are at the research level, not in commercial level. Of course, sex semen is a issue, and we need some sex semen production in our country if we can, but it's very expensive technology because of the patent. But if you look at the long term, I think still it's a worth investment to do. Of course, the ultrasound scanning based PD plus using kits is necessary. It's not that expensive to use ultrasound scan. If I say we don't need big, very sophisticated scanners, but there are cheaper versions available, which I'll show in the next slides. Of course, we need to address the negative energy balance with proper nutrition management. In order to achieve this, we need to, get collaboration between the institutions, between the disciplines, between the people. We need to get out of our, red tapes, bureaucratic, the barriers, and we need to work as one good team. Otherwise, we'll be in a big, because things get more aggravated with importation restrictions, etc., etc. So we need to address these kind of, Of course, ovulation synchronization protocols or programs need to be expanded. There are certain inherited issues of drug shortage, etc., as far as I know, but we need to again address this issue as well. And also, I highly recommend to have on-site sperm testing. Again, people will say, okay, it's not possible because you need some equipment, but it is not now. We have even app-based, very simple tools and techniques available, where even a farmer can easily handle this using his own mobile phone if you have a smartphone. And again, semen quality-wise, of course, we are using the slow freezing technology in the Sri Lankan frozen semen production, but definitely we need to move for the vitrification, where the quick freezing that will preserve the semen integrity and the viability in a great extent than the slow freezing. And like I mentioned, we have to address this endometritis and mastitis issue very seriously with a collaborative way in order to have a proper a few of the examples which I mentioned. Even now we have this, app-based smartphone versions available for the scanners, which are cheaper than the things that we use in research. Obviously, something need to be seriously considered and need to invest from the government side, at least at the VS office level. We need to make these. Of course, we have developed our own technologies, even using ultrasound scanning, just to even, the without any personal bias, you can just detect the pregnancy. And we are happy to share this kind of information also if somebody's promoting this in the country. And so this is one great thing if you can use the semen analyzer on site, on farm, which is phone-based, app-based, small tool, not expensive, very cheap, where even you can slide on your hand with this small gadget, like a small lens. With the app, you can download and then you can go ahead with whether your semen store is good in quality before you do the AI at your own farm. So overall, if you want to have a better reproductive outcomes, good performance in terms of reproductive management, we need to encompass all this genetic selection, nutrition management, disease control, plus the management of reproduction using the 4 key areas which I mentioned. So just let me briefly touch upon the other species in the country, because this is farm animal beings, not only cattle. Obviously, for the small ruminants like goat and sheep, we do not have much issues because we don't have big industry in the country right now. We have little level or small scale reproductive management programs like synchronization going on. Some AI is going on in large scale farms, but not that popular among the farmer level at the small scale level. But it is, again, if you want to, this is again, like small ruminant is one key aspect if you want to improve the animal industry in the country. So with time to come, definitely we may have to focus more on this segment. In terms of swine, again, we don't have serious issue in reproduction wise. But of course, lack of breeding material is a huge thing that farmers are facing. So what we can suggest is, of course, improving or popularizing the AI programs. We have trained many people, many large-scale farms in the country, and they are doing really great. They have really good their own breeding animals with them, and we have introduced the semen processing technology, semen preservation technologies, and of course, going even one step ahead, we have introduced a very low-cost semen extender as well, which is one good solution because imported extenders are very expensive. So that you can easily use these locally made extenders if you want to popularize the pig AI in the country. So that's all I have to share right now in terms of reproductive management. I think after me, Professor Indunil will talk a bit more on some of the other aspects. And if you have any questions on what I have shared with you, I'm really happy to answer.
Thank you very much, sir, for sharing the recent knowledge and experience in the field of animal reproductions and biotechnology. And the audience, if you have any questions, and we have another chance to raise your questions, and you can prepare your questions, and we will meet at the questions and answers session. So with that ones, I would like to move to the next key presentations of the forum. So I would like to welcome Professor Julian Hanley from South Korea. So Professor Julian Hanley is a professor in animal molecular genetics, the divisions of animal and dairy science, College of Agriculture and Life Science, Qtn National University, South Korea. Professor Lee is one of the pioneers in the field of animal genetics, especially poultry genetics and breeding. So Professor Lee pursued his bachelor in agriculture, majoring in animal science from Qtn National University, and also masters in agriculture, majoring in animal genetics and reproductions from Qtn National University. And he obtained his PhD in animal molecular genetics, University of Sydney, Australia. And he joined as a postdoctoral research associate in the Department of Animal Science at the University of Illinois, USA. And also he served as visiting research fellow, the Department of Animal Science at the Kobe University, Japan, funded by the Japanese Society for Promotion Science. Now also he joined the visiting research fellow in the Department of Pathology at the University of Michigan, USA, and working as a Professor Douglas Smith for the swine leukocyte analysis. He joined Qtn National University in 2002, and he he started his career as the professor in animal Department of Animal Science and Biotechnology by supervising more than 10 PhD students and 20 master's students, including the international students from Bangladesh, Indonesia, Sri Lanka, Nigeria, Mongolia, and Uganda. Professor Lee has diverse interest in research fields, especially diversity analysis in Korean native chicken, and studies for the QTL and GWAS for the Korean native chicken, and selection signature studies in Korean native chicken, and the disease resistant studies, especially focusing on major histocompatible region. Professor Lee has contributed his expertise through the different the projects from 2003 to up to now with large-scale projects. For example, BioGreen project, development of economic traits genes from Korean native chicken using the proteomics, and also FTA project, research on the productions of high-quality chicken for the domestic poultry genetic resources, and also the FTA project for research on the development of domestic breeds using the national and private seed and the enhancement of the marketability. And recently he finished the one of the big projects in next generation's BioGreen 21 projects, research on the genome selection technology for development of high quality meat breeds. And also he has several other projects like a GSP Golden Seed projects with several steps to develop the Korean native chickens for the commercialization. With this, the introductions, I want to invite Professor Julian Hanley from the Qtn National University to talk about the trends and opportunities in animal breeding and genetics to meet the global food demands. So welcome the professor.
Okay, thank you very much for the good introduction. Can you hear me?
Yes, Professor. Yeah.
Okay. It is my great honor to give a presentation today, and especially Dr. Manjula. Today, I will talk about the trend and opportunities in animal breeding and genetics to meet the global food demand. Actually, I mostly talk about the Korean native chickens, and I'll talk about the Korean situation and the progress that I carried out last 10 years. Okay, I will talk about the one of the paper published in 2000. I think it's 2019, yeah. Frontiers in Genetics, talk about the USDA blueprint for animal genome research. As you know, the USDA is one of the government organization in the United States. So they cover the whole world actually. So they made 4 goals. Goal 1 is providing nutritious food for growing human population, and improving sustainability of animal agriculture, and also increase animal fitness and improving animal welfare, and also meet the consumers' demand. These 4 goals, they designated as a blueprint for coming 10 years. And I think this forum is good point to share these kind of importance goals together. And in terms of the poultry section, USDA, the National Poultry Research Center, which is located in Georgia, and they gave a couple of the keywords, which is immune related, also some of the meat safety, and also disease resistance. These are the keywords for we have to be handled in the future. As the Dr. Manjula told about the my background, last 10 years I will talk about the mostly the chicken project, covering the domestic chickens. And is there a problem with the screen share?
Yes,
Can you
Professor. The slides are not moving.
Can you see the screen? No,
Not in the presentation mode, sir, Professor.
not in the presentation mode.
Yes.
I will do it again. Can you do that? Can you see that? This?
Yeah, yeah.
Still the same?
Yes.
Anyway, can you see my slide?
Yes, Professor.
How about this slide? It's okay?
Mm, yes, yes.
How about this? It's better?
Yes, Professor. We can see.
It's okay now? It's okay?
Yeah, we can see, Professor.
Okay. I will go further, right? Go proceed. Basically, these are the topics that I carried out the last 20 years. And when you look at the keywords, economic trait, domestic breed, and high quality meat breed, and genetic markers, and also developing of the native breed and immune and disease resistance. Previously, I hear the Dr. Silva's the talk, and animal genetic resources are very important. I think the local genetic resources have kind of very good characteristics of this high quality meat trait, also disease resistance as well. That's my research focus so far, and I think I will do further in during my research period. Okay, the presentation outlines, this is mostly I done last 20 years, and I'll look at the diversity analysis and QTL GWAS. And most of the audience of this talk is the student, and the QTL is quantitative trait focus, and GWAS genome-wide association studies using the Korean chickens. And also I look at the selection signatures in the local breed. Also, I look at the MHC diversity study in chickens. Okay, the first one is the diversity analysis in Korean native chickens. Dr. Silva also mentioned about the mtDNA D-loop variation. There is a highly variable region of mitochondrial DNA. This is very good for the population studies. Also, I look at the mitochondrial DNA D-loop sequences in Korean native chicken, also Smp-based diversity analysis. These are the diversity analysis using mtDNA D-loop sequences. I don't like to go to details, but we found some haplotypes, new haplotypes, and then new there's a relationships with the other chicken breed, which is very specific for the native chicken breed. So when you go to these kind of papers, then you can find out more details. Also, the Smp-based diversity analysis, we use the 600K high-density Smp panel. This is a bit expensive, but we have a lot of information at the same time. So using this
Excuse me, Professor.
panel,
Professor, yes, the presentation is not moving.
Not.
The presentation still is not moving, Professor. So.
Not moving?
Yeah,
you mean?
slides are not moving, yes.
I think it's the sharing the slide problem. Can you see this?
Now,
Maybe, yeah, you have to go to presentation mode, I guess.
Presentation mode.
Can you see this?
yes, but it's not in the presentation mode, right, Professor?
I think the presentation modes have some problems, so I will write this. Okay.
Okay.
Okay. I use the 600K high density SMP panel for these population studies, and also look at the LG decays, LD decays. LD means the linkage disequilibrium. So different population have a selection pressure, then have different LDs. And also using this SMP panel, we use the chicken diversity analysis. Next one is QTL and GWAS, and I will we designed the QTL population. This is called the QTL mapping population, 3 males per line, and mate to 4 to 5 females per male. So we mate together and then generate about 600 chicken populations. This is a resource population for our QTL study. So we generated, at that time, this Smp chip was not available, so we used the microsatellite markers to generate the maps, and then connect to the trying to find out the phenotypic trait. We measured the 91 phenotypic trait to look at the genomic locations of this trait. So this is one of the example, and the meat quality and body weight trait. And whenever you look at the above the red line is a significant region of the trait. So we found out number of the significant locations in the number of the economic trait. And this is also very interesting. The Korean native chicken breed have shank color variation, but there's a lot of variation. So we measure this color variation with the spectrophotometer, and then we identify the L and A, B values. So when you look at the heritability, it's quite high, which is more than 70% of heritability. So which indicate the there's mostly the causative genes can be identified using this heritability and these QTL studies. So we identified in the chromosome 24, there is a very big peak which located the shank color variation QTL located. So previously the other scientists identified the BCDO2 gene, which is shank color, the yellow shank color, affecting yellow shank colors. There's also, we identified the same location. So we published this result as well. And also during this study, we need more like kind of SNPs. And then we generate next generation sequencing data for our Korean native chicken rice. So we identified more than 4 million SNPs at the same time during our study. So this is all this information is deposited in the gene bank. And now recently we generated new populations for our genome research. So in chicken, so we generated the new QTL populations. This is a simulation study, how many animals we need for our study. So basically through this simulation, which is we put the heritability 0.5, and we think the total 35 QTLs located in this or in a certain trait. Then basically 100 animals there we can't find any synchronous SNPs in the during this simulation study. But if we increase 500 animals, we identify 2. If we increase 1,000 animals, we can identify 5 different SNPs for our study. So based on this simulation study, we now generate more than 1,000 F2 chicken populations for our future research. Now I will moving on the selection signatures for the Korean native chicken lines. Selection signatures, these are Korean lines, Korean chicken lines, and these highlines is egg laying layers, and these curved and rows, these are the broilers. So when the people select the layers or broilers, then the genomic region also selected. So which region has been selected for these regions for during the selection procedures? Okay, because we did the Smp chip analysis, we initially general diversity analysis we done. So these are the Korean chicken lines, and these are the broilers and layers, which are very different in terms of diversity. And then when you look at the Korean native chicken and broilers, and these one of the genomic regions, and there is a high red means there is a high linkage disequilibrium, and also it's a failure than this Korean native chicken. So Korean native chicken have more selection than the broiler lines. We identify this kind of regions, and then trying to find the what kind of genes underlying this selection has been carried out. Also, we look at the Korean native chicken and layers. You can see that there is a differences. Also, we are trying to find the underlying genes. Okay, this MHC diversity study in chickens. MHC means major histocompatibility complex. So this gene region is mostly determine the self and non-self antigens. Also, this gene region is contribute to immunity. There's a lot of people investigating this region, but still there are some lack of the informations. In different species have a different name of the MHC, which is human MHC called HLA, and the chicken is a GAGA MHC or B haplotypes. So we are looking the chicken MHC because the local chicken breed have more kind of immune, the disease resistance and very high immunity compared to the commercial breed. That's our assumption. So we're trying to find a lot of information from this kind of MHC haplotypes. Okay, basically the Korean chickens and Bangladesh chickens, later the Samsul Bhuiyan is one of the professors in Bangladesh. He will talk about a little bit about their study, and Sri Lankan chicken populations, which is Dr. Manjura mostly investigated, and some of the commercial chickens as well. So also, we received the standard chicken MHC haplotypes from the USA. So these are the resource populations that we used for this MHC study. Basically, at the initial stage, we look at the microsatellite markers in the MHC region, which is the name is ADI 0258 marker. So this marker is very highly polymorphic, and then we're trying to look at the diversity. The diversity of this marker is mostly located in the Asian population and African population. North American population, which is highly commercial breed, they have less diverse MSC. This ADI marker diversity than Asian and African breed. We think this one microsatellite marker is not enough, so we generate a number of microsatellite in the MHC region. So I'm not going to details, but when you look at this paper, then we identify the number of a lot of information. Also, this MHC region, the French people and Dr. Fulton from USA, they designed the Smp panels, which is called Smp panel 1 and Smp panel 2. We share the information together, and then we trying to identify the MHC haplotypes using the Smp panel 1. So there is A, B, C, different haplotype clusters we identified in the whole population. And also using the Smp panel 2, we identified a lot of new haplotypes in Korean native chicken. I think also your Sri Lankan chicken population also have a lot of population as well. So it's kind of reservoir of the, I say genetic resistance, disease resistance in the local chicken populations. This is a publication we recently publicized in the journal. Okay, it's ongoing research. These are the customized Smp chips. Customized, we designed the genomic prediction of Korean native chickens. We make ourselves the Smp chips to the Korean native chicken genome research. This is a procedures how we can make. And previously 600K chips and the 60K chips available. And then using these two information, and we add up the chicken QTL information as well. So we designed the new chicken Smp chips for our chicken population. Using these chips, previously I told you that I did QTL studies using some of the meat quality trait and growth trait. And this is shank color trait. We identified this kind of new SNPs in that the significance regions. Also, this growth trait we identified recently. Also, this is a paper published in the in 2019, and Stefan Wagen is a German scientist. They developed the synbreed chicken diversity panel, which comprise about 174 different chicken breeds in the world. So we received the data from him, and now we analyze it together with our Korean native chicken lines. This is a synbreed population comprised of 174 breeds. And I think the Korean chicken breed is somewhere here. So we have a lot of data, and then we're trying to analyze ourselves for the world population. And recently, we think using the microsatellite marker and SNP markers is not enough. So recently, we trying to generate the whole region of MHC in chicken using the next generation sequencing the technology. So we generate the whole sequences of this region and using the NGS technology. So this is the overview of what we generated, and then where the genes located. And also these are the NGS sequence mapping result to the reference genome in the database. So this, I think you can see that this is one single dot is one single sequences. So we generate a lot of sequence trying to get the result. Okay, these are the research that we did in our laboratory for last 20 years. And I think the research is still going on in this area, but I think we need more like a collaboration in the future for doing more research together. For last 10 years, I have focused on the animal breeding and genetics, especially in chicken, and still working on the keywords. So there's a number of keywords that I told about, Korean native chicken and diversity, SNPs, and QTL, GWAS, and MHC. These are my main working area. And for the global demand, native chicken breed are becoming more and more important for doing research more. And I told you at the beginning, the USA blueprint for animal genome research. But in terms of the chicken area, chicken breeding companies, they're doing a lot of work. But the problem is the details is confidential. So we have to generate to develop our own populations by ourselves. So a lot of collaboration is needed. Okay, I'd like to thank to funding bodies. Also, I'd like to thank to a lot of collaborated in the world. And also, I'd like to thanks to special thanks to Dr. Manjula of our Wellness University in Sri Lanka, which is the audience here, and also Tisrini. She's main working force in doing MHC study in my laboratory. Also, we are quite international laboratory in Korea, and any kind of suggestions and collaborations welcome from your side. Okay. Thank you very much. Thank you very much. And I'm sorry for my slide sharing, but hopefully you can a little bit understand of my talk. Okay. I'd like to take some questions.
Thank you very much, Professor, sharing the vast knowledge in the field of poultry genetics. We take the casework of studies of the Korean native chickens, and we hope we can join in the panel discussions. If someone has questions, please bring your questions in at the panel discussions. So with that one, I would like to move to the next presentations of the forum. The next speakers of the forum is Dr. Hiruni Vijaysenas, one of the alumni of the OLS University. Dr. Hiruni Vijaysenas, born and raised in Galle, Sri Lanka, completed her education at Sacred Heart Convent, Galle. She completed her bachelor degrees in the field of animal science from OLS University of Sri Lanka. She received her master's in animal breeding and genetics from University of Saskatchewan, Canada, and completed her PhD in swine genetics from University of Nebraska, Lincoln, USA. Currently, she is working as a postdoctoral fellow at the USDA Meat Animal Research Center. So today, Dr. Hiruni Vijaysenas is going to talk about applications of advanced genetics and genomics tools in swine productions. Dr. Sudhini, over to you. Dr. Hiruni, over to you.
Thanks. Can you hear me fine?
Yeah, we can hear you.
Okay. Is the screen looks good too? Can you see my screen?
Yes, we can see.
Okay. Yeah, thank you for the introduction, Prabuddha, and thank you very much for inviting me for this forum. It's an honor to come back to my university as a guest speaker and share some of the things that I learned during my graduate studies. So today I'm going to talk about how we can apply advanced genetics and genomic tools in swine production. So when we talk about swine reproduction, the success of a breeding female largely depends on the reproductive longevity of that animal. We usually measure reproductive longevity as number of lifetime litters that female produce, and also number of lifetime piglets that animal produce. So it has been shown that a female has to at least produce 3 litters during her lifetime to recoup her development and maintenance cost. So swine breeder only start to make a profit from a female when she starts producing her 4th and 5th and onwards litters. So her first few litters, the money she makes from the first few litters always goes back to recoup that development cost of that animal. So about 40% of the replacement females that we use in swine industry don't meet this requirement. Most of those animals don't even get pregnant at least once, or sometimes they get pregnant once and then they farrow, but they never return to estrus again. So this is a major issue in the swine breeding industry, because when these animals are not performing, we have to cull these animals and it becomes a welfare issue, and it's also added cost because then you have to replace those females again. So, like I said, the major goal in swine reproduction is to improve reproductive longevity and efficiency of these breeding females. Some of the main reproductive traits that we try to improve are listed here. So first one is age at puberty. It has been found that age at puberty is an early indicator of reproductive longevity. If an animal reaches puberty early, that animal has a tendency to stay in the herd longer and produce more litters. And then we also try to improve litter size traits like total number of piglets born, number of piglets born alive, and also we try to reduce number of stillborn piglets. And then, like I said earlier, we try to improve number of lifetime litters that an animal produce, or we call it reproductive longevity. And then we try to improve the teat number and also weaning to estrus interval. That means from the how long does that animal take to return to estrus after weaning her piglets. So we want a shorter interval during this time. So we know these reproductive traits are really important in swine industry, but there are some challenges to improve the reproductive traits. One major challenge is that these reproductive traits are mostly lowly heritable. That means they are largely influenced by the environment. So if you remember the equation phenotype equals genotype plus environment, these reproductive traits are largely influenced by environment. So the genetic portion is pretty low. The genetic portion that contributes to phenotype is pretty low. And also, most of these reproductive traits are expressed late in life. So at the beginning of an animal life, these traits, phenotypes are not available to the swine breeder to do phenotypic selection. So to overcome these issues, one major solution is to use genomic selection. That means you can use genomic approaches to identify genetic variants that can predict a genomic estimated breeding value for that animal at an early age. So without even having phenotypes for reproductive traits, at a very early age, you can predict which animal will be better at reproductive traits, and you can select those animals for breeding. So over the years, like past 20 years, I suppose, like they have started developing different genomic approaches and genomic tools to accomplish this goal, to identify genetic markers that can predict the future reproductive capacity of a female. So these are some of the main genomic approaches and tools that we use nowadays, like develop DNA marker panels, or we call them single nucleotide polymorphisms, or SNPs, and do genome-wide association, candidate genes and variant search, and also next generation sequencing, mainly whole genome sequencing and RNA sequencing and gene expression analysis. I'll talk about these approaches very briefly in the next few slides. So the first one is DNA marker panels. I think the first swine DNA marker panel was developed in 2009. So here what they do is usually like different commercial companies, different research labs identify a lot of DNA markers that are span across the swine genome, and they incorporate these DNA markers into marker panels that look like this. I'm not going to talk about how these are manufactured or how the genotyping works, but this is a very popular tool at this point era. And there are lots of different types of marker panels available. Some are like trait specific, some are just generic markers that span across the genome. And also the marker density varies widely. Some has like 50,000 markers, and it can go up to like 100,000 markers. And then you can genotype each of your animals for all the markers in a marker panel. So you can use one of these panels for each animal and genotype those animals for each marker. And then you can use those genotype data to do genome-wide association analysis. So here what we try to do is try to identify associations between genetic variants with the phenotype of interest. So you genotype your animals using those DNA marker panels, and then you also have to collect phenotypes for the targeted trait from your animal population. And then you use different statistical approaches to predict the associations between genetic variants and the phenotype of interest. So this is an example of a genome-wide association that I did during my PhD for number of lifetime litters produced in swine. So here, different colors represent a different chromosome in the swine genome. And each of these dots represent a DNA marker that we have in the marker panel. So what this graph shows is that these high peaks that you see in these 4 chromosomes, that means these genomic regions are significantly associated with the phenotype of that I looked at, in this case, number of lifetime litters. So I found 5 genomic regions in the swine genome that are significantly associated with this trait that I looked at. And then once you identified those significant genomic regions, what you can do is you can go and explore that region more and scan the region to identify if there are any genes located in these regions, specifically genes that are associated with a trait. So if you think there's a gene that's highly associated with litter size traits, you can focus on that gene and try to identify if there are any other polymorphisms or genetic variants in that gene. So what you can do is you can do a PCR and then sequence that gene and try to identify any other genetic variants. For an example, this is a sequence of a particular gene in 2 animals, and at this point, you can see a genetic variant or polymorphism in that gene between the 2 animals. So this is important because those DNA marker panels that I talked about don't include genetic variants from the entire genome. Those are just subsets of DNA markers that you find in the genome. So these don't include all the markers that you can see in the genome. So this way you can identify other polymorphisms that can be associated with the trait that you're looking at. So those are some of the very common tools that we use nowadays. And there are some other developments like next generation sequencing. So the first one is whole genome sequencing. So if you remember the central dogma, so we have DNA, and then DNA is transcribed to RNA, and then RNA is translated to protein. In this case, what we try to do is sequence the entire genome or the DNA sequence of a pig. So in swine, the swine genome is about 3 billion base pairs long, and we sequence the entire swine genome. The main advantage of whole genome sequencing is you can identify a lot more genetic variants that are not included in those genotyping marker panels. So I did whole genome sequencing for my PhD, and we found about 1 million genetic variants span across the genome in our population. And so you can do more genome-wide association using these DNA markers and try to identify genetic variants associated with your trait. Another main tool that we use is RNA sequencing. So that's the second step of the central dogma. And here, so we know that RNA is not expressed in every tissue every time in every individual. So RNA expression is dependent on age, specific body conditions, specific disease conditions, likewise. So, so this is a very good tool to measure gene expression differences between different sets of animals. So what you do here is you pick a tissue that is important for your trait. So if you look at litter size traits, maybe ovarian tissues or uterine tissues would be a good tissue. And then you collect those tissues from two different animals. So if you have a set of animals that have smaller litter sizes and set of animals that have large litter sizes, you can collect tissues from those two groups, and you can compare the expression of RNA in those two sets of animals in your tissue, like ovarian tissue. And you can see if you can see different differences in gene expression. So if there's a gene that you think is really important for litter size traits, and if you see differences in expression of that tissue, of that gene in the two different groups, you can confirm that maybe this gene is really important for litter size traits, and then you can characterize that gene further for genetic variants. So that's the main advantage of using RNA sequencing. And so most of the research groups and commercial companies always try to integrate all these genomic approaches to increase the power of their genomic analysis. So they just don't do one genome-wide association and stops there. So this is an example of how we can integrate those genomic approaches. And this is a part of my PhD research. So I actually used all those approaches that I talked about. I used genome-wide associations, then RNA sequencing and genome sequencing to identify genetic variants that are associated with reproductive traits. And then I incorporated all those genetic variants into a DNA marker panel for reproductive traits. So this marker panel includes about 100,000 SNPs. And now we use this as a, in the commercial industry to select for reproductive traits. So since this is a breeding and reproductive forum, I mostly talked about reproductive traits, but these genomic approaches I talked about are widely applied for other economically important traits too. So one main trait is disease traits. So these are some of the common swine diseases that we have here, like PCV2, PRRS, and pestide virus. And so host genetic variation is known to play a role in susceptibility and diversity of disease development. So if we can identify genetic variants associated with disease susceptibility, then we can help to select animals that are less susceptible to diseases for breeding. And this can actually help to reduce costs associated with vaccines and pharmaceutical products. So this is a genome-wide association that we did for PCV2B, and we found two large significant genomic regions associated with PCV2B in swine. And in addition to identifying host genetic variants, we can also sequence novel pathogens and like viral pathogens, bacterial pathogens, and we can characterize those using this nanopore sequencing technology. This is a very small sequencer that you can even put it in your pocket. So, and you can just connect it to a computer and sequence an entire viral genome in real time, and it's really fast. It's amazing how it works. So these kind of techniques can help you to identify novel pathogens and genetic variations between different viral strains. Using this technology, we identified a new pestivirus in our population that actually caused shaky leg syndrome. In addition to disease traits, you can also use these genomic approaches to improve body conformation traits, because we know if the female looks good and if the female has a really good structurally sound, then that animal can be a better breeding animal compared to a weak animal. So we always try to improve their front and hind leg conformations and the back quality. We always try to select animals that don't have foot lesions, likewise. And then meat quality traits. So in here, I'm actually stepping into unfamiliar territory because I'm not by any means a meat scientist. So I'm not going to talk a whole lot about meat quality traits, but I know that we use these genomic approaches to improve meat quality traits like intramuscular fat, water holding capacity, marbling score, meat color, and pH. Okay, so to summarize this presentation, like genomic selection has become an integral part of swine breeding programs. Basically, all the swine breeding companies use this genomic selection nowadays to improve many economically important traits globally. And so the ultimate goal of many swine genetics research is to identify genetic variants that can be used by farmers to select the best animals for breeding at an early age. So basically use genomic approaches to calculate genomic breeding values and improve genomic selection. One major issue with these genomic approaches is that at the moment they're very expensive. So you need a lot of funding, research funding to conduct these experiments. So there has been lots of research done, continuing to be done to find ways how to minimize the cost associated with these approaches. And with that, I'll finish my talk with one of my favorite pig quotes. Thank you.
Thank you very much, Dr. Hiruni, for your wonderful presentations, and we hope you will join with the discussion session. So with that ones, I would like to move to the next presentation of the today forum. So I would warmly welcome Professor Indunil Pathirana. Professor Indunil Pathirana is a professor in animal science and veterinary surgeons in the Department of Animal Science, Faculty of Agriculture, University of Pune, Sri Lanka. Professor Pathirana received his bachelor in Bachelor of Veterinary Science, University of Peradeniya, and his PhD in the field of reproductive physiology from Osaka Perpetual University, Japan. Professor Pathirana completed his postdoctoral research in the University of Sydney Dairy Research Foundation, Canberra, Australia, and also in the Osaka Perpetual University Laboratory of Serology, Osaka, Japan. Professor Pathirana has several awards, fellowships, and honors, especially the President Award for the high impact scientific publications in 2015 and 16. And also, he received the Endeavour Leadership Awards in 2018 in Australia. Also, the Japan Society for the Promotions of Science Fellowships in 2013. He also received the Travel Awards to attend the conference in Australia, USA, and India. Professor Pathirana's the research interests are focused on animal reproductions, animal andrology, and animal productions. And also, currently he's working on use of Progesterone enzymes, immunoassay for pregnancy diagnosis in Asian elephants, and assessment of heat stress in dairy cows through the quantifications of heat shock proteins, and also improvements of the success rate of artificial inseminations in Jamnapari crossbreed growths through the estrus synchronizations, and also studying the hormonal dynamics of selected, domesticated, and wild animal species during the male sexual development. With that one, I would warmly welcome Professor Indranil Pathirana to talk about the advances and limitations in cattle and buffalo reproduction in Sri Lanka. Professor Pathirana, over to you.
Yeah, Prabhuddha, hope you can hear me well.
Yes, sir, we can hear you.
Okay, I will share my slides.
Yes.
Is it full screen now?
Yes, Professor.
Okay. Yeah, thank you very much for inviting me for this forum. And always good to see that this young energetic group of lecturers from Ovulation University is thinking about the field of this livestock, and also supporting the students and giving a flavor to the students for this, regarding this industry actually. So that is actually, I really appreciate that our students should have a flavor about our, the field situation. So this is one of the best opportunities in my opinion for your students. I saw that many of these students are attending, and so I will focus on this the given topic, advances and limitations in cattle and buffalo reproduction in Sri Lanka. And since Professor Suranga Kodituaku touched cattle reproduction in detail, I will not going to, I'm not going to focus on cattle reproduction much, and it is really a kind of opportunity for me since he discussed many of the things in detail. So I'm actually mainly focusing on the real situations, real situation in our, under our field situation actually, and also to give an idea about our field situation to our, the students. And no matter, these students are coming from Ovulation, Ruhuna or Peradeniya, I thoroughly believe that they should have an idea about the field situation. So this is a real good opportunity and a good idea to have this kind of, to give an opportunity to these two students. So, as far as I believe, this management of reproduction is based on a simple fact, that is a calf per year. So, irrespective of the efforts we make, that the simple thing is that a calf per year. So we do everything to achieve this target. As far as I believe, we have a real problem in the, the field. We have to correct this issue. And in my opinion, the limitations we have to develop the field of reproduction or applications of reproduction is a problem of this nutrition. I think Professor Suranga Kodituaku also very correctly mentioned that I think that we should correct this problem, which is really making troubles in the field. And if we can overcome this problem, if we can provide enough nutrition to these animals, then we can talk about advances from this side of reproduction. If we are unable and if you are going to struggle even in the future with these nutritional issues, definitely we have to talk about these limitations for a decade or, several decades. So although I'm a reproductive scientist, I still believe that 80% of the Sri Lankan field situation is suffering from these nutritional issues. So this is a kind of a reflection of this field situation. With this kind of situation, what is the advancement of, we can think of? Just get an idea about these real figures. So the the second column is the data we obtained from a dry zone farm. Actually, at that time, Ridiyagama farm, they maintain a herd of relatively, big herd of buffaloes. Mainly, they were Niliravi and Mura crossbreeds. And in 2014, we went through records. Actually, the problem in buffalo sector is that, after these kind of legendary or senior scientists who were there in Peradeniya and also Ruhuna, like Professor Oswin, Professor Abegunardana, Professor Ibrahim, and then Professor K.K. Patirana, and then Professor Singh. And after these pioneering scientists, no one to look after buffaloes now. So we do some kind of basic things, and actually not to neglect this group of animals. So just see these data. Age at first calving in 2014, it is almost 5 years. Then after 6 years, in that is Marandavila farm, close to Bingiria, again 5 years. So irrespective of the climatic conditions, no change in age at first calving. So we are talking about these advances in this background. Calving interval, see nearly almost 2 years, at least more than 1.5 years in case of Ridiyagama farm, which is no more now, a kind of buffalo farm. Now it's a dairy farm. And again, although Marandavila relatively, they maintain these buffaloes relatively better than Ridiyagama, still it's nearly 1.5 years, right? Although it's not very bad under true field conditions. So we are thinking about this advancement with this background. So I'm sure that although we are thinking about these advancements, our real field conditions are not ready to accept those. So my point is that we should correct this first at field level, which is not so difficult. So I'm going to discuss about this maybe 4 to 5 year short-term plan here, totally for the benefit of the students who are joining today. Okay, so then we are proceeding with a set of limitations. What are these? No argument, we all can agree that low plane of nutrition is a problem. Definitely, if we assess BCS, body condition score, definitely low and not up to the desired level. Without correcting this point, in my opinion, even Professor Suranga mentioned that no point of applying these advanced techniques. So it's our duty. It's a kind of collective, should be a collective effort to correct this point, right? So then the environment and heat stress. If we cannot provide ad libitum water to these animals, what are the advancement we are talking about? So that is not so difficult. We all know that there are practical problems for some of these farmers, but still, if we emphasize the importance of providing water, I think these farmers will follow us. So we import animals from countries like Australia, New Zealand. Do we, concern about the environment we are going to manage them? So heat stress is also a problem here, and overall the environment should be at least favorable. May not be comfortable, but at least should be favorable. We always talk about this importation of animals and about heat stress. Do you believe that in Australia, if you specifically talk about Sydney, during the summer, they reach, not actually reaching, but sometimes they pass 45 degrees centigrade, at least 4 or 5 days per season. But still, these animals are high producing. Only very minor effect on production, totally because of nutrition, nutrition, not the genetic makeup, in my opinion. So then another issue that we do research, but in my opinion, those are individual and isolated. And we are collectively responsible. Actually, all these researchers, those are not diffused, to the field level, not handled at field level. So I would say no, or a few field applications. We cannot blame others, should be blamed ourselves. Only a few, a group of people, even I can read names, less than 10, who is dealing with reproductive research in Sri Lanka. I'm sure that Suranga is also well aware that only few of us are doing, research. So, it's our responsibility, not the previous generation now, to get together and transfer this possible technologies to the field level. So we should be blamed, right? And then the availability and cost of hormonal drugs, provided that we fulfill the previous limitations and we overcome those. And there is a set of another issues coming. I'm sure that we can list a few. Okay. Then how about a better situation? Imagine just, we will go through this. If we are organized with, at field level, can't we, not all, I'm sure we cannot educate all these farmers, and even we cannot influence them for these applications, not 100%. We can proceed, maybe 20-30%, and with maybe 1 or 2 years, maybe we can reach maybe 50-60% So then provide ad libitum water, year-round uniform feed supply, and then how about the favorable environmental conditions to these animals? And then then we can think of these assisted reproductive technologies and possible applications. So if limitations exist, we cannot talk about this ART. So then we have to correct these problems first at field level. Then we should get these technologies to field level. And I'm sure that all of us, I mean, at least we do have few good findings. So just imagine this kind of situation. This is not far away. No reproductive technologies, no ART at all. So this is actually one of the ideas from kind of active dairy veterinarian from NLDB. Yeah, we work together, and Dr. Samira. So he took 145 non-pregnant heifers, including some, heifer calves even, from Marandavilla to Singapata farm, just 50 kilometers away. So he gave this comfortable or at least favorable conditions to these buffaloes with the help of the management. So maybe reared for nearly 15 months, gave these conditions. Do we believe that now 93 of these animals are pregnant? No hormonal treatments, nothing. So minimum concentrate feeding, right? So this kind of condition for buffaloes. So I'm mainly focused on buffaloes because Suranga discussed enough about cattle. So then we tried one synchronization protocol. We call that actually a modification to OviSync, which Professor Suranga mentioned previously. Application of SIDA at the beginning, once we gave this fertirelline. Then after 7 days, we removed SIDA, one clopastrone injection, and then after 2 days, another fertilization injection, and then timed AI. So we all know that if it is possible to have a timed AI in buffaloes, it would be an added advantage. So actually, this is a kind of preliminary study. We conducted this at one veterinary range, Kahavatta, and then again at Ridiyagama, long time ago, at least 6-7 years ago. And then we compared this protocol with well-known prostaglandin double dose regime. So actually, without any improvement or enhancement in plane of nutrition, so this treatment gave nearly more than 50% of the success rate compared with the PG double dose, which is widely used in the field. So everyone is using this PG double dose regime, right? But important point is that this is really costly. I mean, SIDA for this protocol, definitely the cost is more than 6,000-7,000 rupees. But if you consider successful lactation and a calf from a crossbred or purebred buffalo, definitely the cost is negligible. So we cannot forget that part also. So then something about goats. So we all believe that it's not very successful to have estrus synchronization in goats. So then we thought of having a treatment protocol with a kind of alternative drug, which is used for human, that is used in women to prevent premature labor. We call the trade name is Prolutin, that is 17-alpha hydroxyprogesterone. Actually, we tried this at Kottukachia and also Halamada, NLDB farms. Yeah, you can, so we discussed this at previous SLVA meeting, and we can provide this information if you need. This is actually should be applicable at field level, right? So we really believe that these kind of work should go to field level. That is why we mainly focus this SLVA, because almost all these clinicians gather there, and low cost for this pollutant it's nearly 800 to 1,000 rupees. We can serve 10 goods using one vial, one ampule actually. But relatively, the way we flush these animals were costly. So I agree. And yeah, we can give information. I am not going to discuss everything here, but we can think of alternatives, future, right? So then, so we measured progesterone and at maybe around 30 days. So we confirmed that pregnancy and also through ultrasound scanning. So just see these findings. Pregnancy rate was 100%. We used 31 dose, all were pregnant. So there were 43 kids, 41 were live, and actually there was intestinal torsion that is not related to this treatment and kind of accidental problem. So that doe, was died, and we couldn't get this, twin birth. So then twin births were 12 out of 31, as a percentage nearly 40. And so there were 11 twin births because of this treatment, and we compared the same period of the previous year in the same farm, they had only one twin birth, right? So we like, we would invite all these clinicians and to apply this and see and to have comments from the field level. And how about the stress, heat stress and reproduction? There's a close relationship. So it may adversely affect HPG axis and thereby the follicular dominance and the suboptimal corpus luteal formation, and end result would be low fertility. And it would be great if we can have a method to assess heat stress easily using a non-invasive sample. Very recently, actually just a few weeks ago, we published this information, and we could detect this heat shock protein 70, which is considered as this is released as a kind of result of inflammatory response in this, these animals. And we detect this in milk, right? So here, during the summer, actually we did this, in University of Sydney farm. So during summer, we observed that the mass of this HSP70 heat shock protein 70, maybe the students, maybe you can refer what is HSP70. And those, levels were high during hot weather compared to the autumn, right? This is not even winter. And then when we consider high yielding animals, based on the per day or per AM PM milking, the calculation, it was not significantly higher, but there was a tendency. But for the milk quarter, it was significantly higher in high yielding animals. So why we should forget male reproduction, male counterpart? Here, we did some research on male sheep, again at Horakale farm, NLDB. Although we have just, maybe 10, 11,000 sheep in our country, not a popular industry, but one of the latest, relatively, I would say, relatively new hormone, insulin-like peptide 3, that is a member of relaxing family of peptide hormones. And we argue that it is a better alternative compared to testosterone to detect the sexual development, where we got kind of starting from 3 months kids up to 2 years, actually 3 years. So there was a gradual increment in case of INSA3, this insulin-like peptide 3, but in case of well-known testosterone, up to 12 months, there was no clear increment in case of testosterone. But later on, we observed observed an increment. But here we believe that this is more reflective. With this testicular development. How about pigs? So we tried testosterone as a initial or potential biomarker for cryptorganism. This is actually not new, but obviously it's new when we consider local conditions in Sri Lanka. We use this as a kind of biomarker to detect cryptorganism, which is a hereditary reproductive disease. We believe so. And but this is a kind of preliminary study using a few, limited number of samples. So we should do further studies using higher number of animals. And also it is interesting if we can measure insulin-like peptide 3, which we claim that better alternative as a primary hormone in case of males compared to testosterone. Okay, suggestions to suggestions to initiate advancements. As I previously discussed, a group of people, leading, with these nutritionists and veterinarians, and also reproductive scientists, and also the government channel, which is responsible for this, should make an action plan, not just a policy, at least as district level. So we should apply this. And then force of trained extension workers would be really helpful, and a follow-up work should be done. Just introducing these techniques would not be enough. So this group of people should be well trained. Existing system, it's not. We can argue that it's not a very healthy situation, right? So veterinarian and his or her staff is specialized for the task, and they are handling the channel. But funding is coming to the divisional secretariat. That is, majority of funds are coming through and handled by, recommended by several development officers who have never exposed to agriculture or livestock. So it's a problem. So these are real field, problems. So then if we correct this, so then we can think of applying ART, and I'm sure that then people like Suranga, and they have done enough, and then we can get together and go to the field level. Introduce these techniques to field level. And final slide. Although this is the theme is different, why we do not apply these reproductive hormones and the studies to wildlife. So the must in Sinhala we call mother kipima. So it's a real problem in case of male elephants. They become really aggressive during this period. And we measured actually, we tried an antagonist, GnRH antagonist, to suppress this must even after appearing this must, right? So then to, based on these, findings, so once we see these must, we injected this drug to these elephants. This is an antagonist of GnRH. We call degarelix acetate. And then all of a sudden, within few hours, we could suppress the must. So imagine, so this kind of endocrinological application, pharmacological application would be critical in case of these tuskers used in Perahara, something like Candy Perahara. These tuskers are priceless. not each and every animal can be used for this purpose. So, and any animal approaching must. So we can use this kind of drug, although it is really expensive, but we cannot forget that the event is also so critical, and there are people who can invest on this, considering the situation. So this is just for your interest, because there are several students more than 80% I observed that students are attending this. So this is actually really important. That's all. Thank you for taking your time. Thank you very much for inviting me.
Thank you very much, sir, for your nice presentation and wonderful time you gave with the information, the slides. Thank you very much.
I think we can all agree to the fact that a list of critical points concerning the livestock and poultry industries and subjects linked to this regard was brought to our attention during the very fruitful technical session that had just been summed up.
Yes, Rasika. From the present status of the genetic potential of Sri Lankan farm animals, a mechanism to utilize the existing gene pool for getting maximum productivity, and global trends on enhancing the productivity of the livestock and poultry industry, we have been made aware of it all. Now, we are ready to open this platform for the panel discussion, where you will be provided the opportunity to raise questions to facilitate the exchange of more knowledge and ideas.
During the panel discussion, your queries related to topic of interest will be addressed. You are able to direct your questions to one or multiple panel members. Once again, we remind all participants that they can either use the raise hand option and verbalize your queries or inquire them through the chat box. Questions posted through the official Facebook page and official YouTube channel of the Animal Science Society shall also be taken into discussion within the due time.
With that, I would like to respectfully hand the discussion over to the chairperson, Dr. Prabhuda Manjula, and the distinguished panel members.
Thank you very much. Actually, we had wonderful times and fruitful times, and we discussed more information regarding the animal genetics, breeding and selections, and the reproduction aspects. So we had distinguished panel members, Professor G. Silva from University of Peradeniya, and Professor Dr. P.G. Selviratna, Director, Animal Breeding, Department of Animal Production and Health, Sri Lanka. And also Professor Surang Kodituapu from Department of Animal Science, University of Peradeniya, Sri Lanka. And also Professor Julian Hanley from Qtn National University, South Korea. And also Dr. Hiruni Vijayasena from USDA Animal Research Center, USA. And also Professor Indranil Patirana from Department of Animal Science, University of Kokoda, Sri Lanka. So let me read the titles we discussed during the past few hours. So Professor Pradeepa Silva summarized the present status of animal genetic resources in Sri Lanka by highlighting the key aspects of the genetic resources in Sri Lanka and how we can use these available resources for the purpose of improving the animal genetics and the reproductions in Sri Lanka. And also when it comes to the Dr. P.G. Selviratna's speech, he highlighted the pros and cons in utilizing the animal genetics resources in Sri Lanka, specifically highlighting the breeding policies and strategies we have in the country, and what kind of practical implications are in the animal breeding and reproductions. And Professor Suranga Kodituapu highlighted the present and future status of animal reproductions and biotechnology, especially how we can improve the reproductions of the farm animals, particularly by applying the biotechnical tools, and what kind of limitations in Sri Lanka to apply those biotechnology tools. And with that one, so Professor Julian Hanley highlight the port genetics, especially with the case studies from Korean native chickens to address the trends and opportunities in animal breeding and genetics to meet the global food demands, in highlighting the what kind of research we can go to characterize the genetic resources of the local chickens particularly, and how we can use those the information to improve the local the chickens, and how we can go for the global food demands with these genetic resources. And then the Dr. Hiruni Vijay apply her the expertise on the field of applications of advanced genetics and genomics tools in swine productions, particularly highlighting the reproduction traits and the genomic selection and whole genome sequencing and RNA sequencing to unveil the genetics profiles that we need to enhance the reproduction traits in the swine, as well as the disease traits and body conformation traits that particularly targeting the scale. And the last but not least, the most important topics from Professor Julian is the advance and limitations of cattle and buffalo reproductions in Sri Lanka. In that case, Professor Patirana highlighting the buffalo reproductions and the correlations between the nutrition and the management of reproductions. So without highlighting the nutrition requirements, we can't go for the reproductions applications. So he highlighted the importance of nutrition in case of improving the reproductions and the biotechnology aspects of the farm animals and the poultry. And particularly the buffalo reproductions and the condition that required for to support the livestock the reproductions. With these topics, we highlighted the key aspect of the animal the genetics, animal breeding, and reproductions in Sri Lanka. So we get kind of the big picture of the country. So now I cordially invite the audience to raise your the questions and your discussions or your the questions to the respective the panel members. So you can send your the questions through the chat box, or you can use the raise hand options to ask the questions. So now the panel is ready for your questions. Thank you. We have one question from the audience. It's a Sunil Gamagre from the audience. Yeah, you can.
Yeah. Thank you. I must commend the presenters. I'm a former Deputy Director of Veterinary Research, retired in 2010. And now we have to translate this into economics, whatever discussed earlier. So a few tips and things to discuss. What I'm giving is that first I discussed this with Professor Pradeepa on the circular 2005 stroke 5. So I already discussed. Other thing is 95% of the farmers have less than 3 hectares of land, and they free graze their cows and produce 90% of the local milk supply. That is one point that we have to think about when we are translating this discussion, the presentations, the new techniques at the field level. They have indigenous and crossbred breeds with a milk production ranging from 1 liter to 15 liters on the average. There may be 20, 22. only 15% of the farmers integrate cattle with crops, coming down from 20% in 2001, Department of Census and Statistics. It has come down from 20 to 15%. But what I have observed is in my studies that these farmers integrating dairy with crop gets 2.5 times more income with crop than those with crop only. And then they produce 40% of the local milk supply. 60% is imported, and these people produce 40%. 20% of the households have indigenous poultry keeping, and 3 to 5% of the households are goat herd keepers. So we go to these are, I'm talking about crossbred, indigenous, and locally. When you talk about genotype, environment interaction, these things. So President Gotabaya Rajapaksa's organic farming became a failure because 95% of the seeds used in crop farming is high input. So our farming requires both high input genetic material and low input genetic material. So this we have to keep in, we should remember when we are translating what you discussed to the farmers. Then indigenous chicken for family poultry, there's a severe shortage in the field level. There's a big shortage. CPRS Karandagolla indigenous chicken project started in 1997. It had 4,500 parents, population, and 12 ecotypes were collected, improved and distributed to farmers, and this was stopped. So now someone had to start something like this, or the veterinary research institute will have to do this. 82% of the farmers are part-time farmers. This is Department of Agriculture census, 82%. So only 18% are full-time farmers. So we are talking about observation of heat and all that with that. Then unlimited quantities of silage feeding during dry period till conception. In a JICA study which I worked, conception levels improved, and silage feeding at growing stage, heifers started cycling by the 20th month. So there are possibilities that we change some of the, in this discussion, how to pinpoint these activities. Other one is goat. The main problem observed is lack of resistance to GI nematodes. It has been estimated that the economic loss is between 57 to 59% due to GI nematode resistance. Professor Velgama's protocol on GI nematode control at an early stage have been very useful in this. This is all studies, this is all practically done with farmers. So you can discuss these things. Then I think these have been there for the last 20, 30 years. Wild boar exotic breeds and Paradenia faculty also did this, have a higher economic value if you look at it. I mean, instead of exotic, the meat value, the meat quality, fat levels, all those things are controlled by this, and we got a very good economic value. Reuse home garden in rural Sri Lanka with livestock. This is, see, home garden. Now, why I'm bringing this to this forum is that this is the place where you can start your work with livestock. Home garden, when we came to farming, before that we were hunters. We were in the jungle. Jungle gave us food, gave us clothing, gave us wood, firewood, everything. What we did was we brought that to our homestead, and we developed a home garden similar to that. And we had cattle, goats, poultry, indigenous poultry, everything there. So this is a place that you can start with in here. Then other one is reintroduce forest free grazing of ruminants to improve biodiversity in forests. These are Europe, they have started this already. We are talking about, our forests are dying. It is dying not because of people are chopping trees. It's because of there's no diversity. ruminants and birds are some of the things. We have lost about 200 fruits, types of fruits, jungle fruits that we have been eating. So ruminants can come into this. And I mean, I can expand on that later. That is one. Then duck, rice, azolla, integrated farming, I did it with MARD project in USAID project in 1991, 1993, and also at the Central Poultry Research Station. There's no way of doing it because department stopped bringing ducks. So these are some of the things that you can discuss in this. Now use that knowledge that was presented by the presenters and show how in this discussion, show how we can bring that to get to make farmers full-time farmers, from 82% part-time farmers to a full-time farming community. And remember, Corona was very helpful for us to think about what's happening. We can't import feed. Now the broiler industry is in trouble. Some of the cattle industry is in trouble. So we have to have the high input production there, as well as the low input production. And another type of production that I call sort of alt system there we can develop in our own farming systems. Thank you very much for giving me this opportunity to give my points. I think the panel should be able to discuss some of these things, how to relate it to farm economics. Thank you.
Thank you very much, sir, for your wonderful ideas and the suggestions to that you bring into discussion at these forums. So we hope in futures we can implement some of the projects with the help of this distinguished panel members and the other institutes, especially state universities who are doing this, the research on the animal breeding, especially farm animal breeding and reproductions of the targeting the poultry as well as the livestock. So if you have any questions for the panels, please raise your hands using the hand options, raise hand option. We have one question to the panel members. So the question is, when preparing the national breeding policy, what are the factors that we mainly consider? Officer Engineer, sir. Okay, so I would like to bring up one of the questions I think that is related with Dr. Julian Hanley, which he can answer this question. What are the best swine breeds to get the maximum profit under Sri Lankan conditions? Dr. Hanley, so we can't hear you.
Sorry. So if you're looking at meat production wise, and under like hardy conditions, maybe Duroc would be a better option. And if you're going for litter size wise and maternal traits, large white and landrace would be a good condition. But also with tropical climates, there might be some issues, since these are mostly temperate breeding. But if we can get like hybrids, that would be, we can take the advantage of heterosis. So that's one way to go there.
Thank you, Dr. Henry. So in that case, since Dr. Pratibha Silva has highlighted that kind of local breeds are available in Sri Lanka. So with your knowledge, so how we can incorporate that genetics to improve the local breeds, like the current the genetics tools and the techniques. I mean, in scales, how far we can go with, at least to initiate the to develop kind of breeds so that's suitable for our purpose.
Yeah, so I think the approach I see there is like, we do have a lot of local breeds. So if we want to introduce temperate breeds, I think the main is the way to go is get hybrids and get the heterosis effect. So that means the cross between the locals and the temperates are going to be better. So genetic improvement wise, so we can start applying some genotyping for some traits that we like to improve and start developing breeding values. I don't know how far we have done that in Sri Lanka. So if it's like a starting point, is it going to be a starting point or like improvement wise? I'm not sure how far we have gone there. So I think the starting point would be to start genotyping at least like few markers, not the marker panel level, but at least if you can do start with like these 100 markers and try to start estimating breeding values, I think that would be the place to start.
Okay, thank you very much, Dr. So,
I think I have one comment.
Yes, Professor.
For use the local breed,
Yes,
especially in pig, and we have to define what the benefit for the consumers. Why consumers need that local breed? So it's probably meat quality or good taste. What kind of any kind of things? Why we use the local breed? Why people like that local breed?
so
And
basically,
that kind of definition, so you have to set up first. And then what's the weak point of the local breed? Maybe the growth rate is very low, right? And then trying to improve. How can you improve that trait by crossing with the commercial breed? There is a number of the companies selling the seed stocks. They are same Landrace and Yorkshire breed, but it's not perfectly same. They have different companies have different kind of breeding goals, so they are all different. So basically, what you like to improve, why the consumers like to have, that kind of definition is first. Then integration to the to the commercial breed. It's not that that difficult, I think. Vice versa, it's not that difficult. It's a couple of generations making plans. It's not that difficult. It's the same situation in Korea, and native breed, which have a very high good meat quality, and taste is better than the commercial breed as well. And it's kind of integration to the local breed. And it looks like this Landrace, but the taste is totally different. What people is doing, the scientists is doing, whenever the crossbreeding of the local breed, and then find out the specific markers, DNA markers, whether they always trace to the next generations. And then it's not that difficult. So you have to find out the solution first. Okay.
Right. Thank you very much, Professor Lee. So, and also I have...
Can I answer the first question asked somebody about the factors that we considered to develop the animal breeding policy?
Right.
Yes. In case of Sri Lankan animal breeding policy, the committee has considered the environment conditions prevail in the different parts of the country. So they have considered the agroclimatic zones. So each and every agroclimatic zones, the relative humidity, prevailing ambient temperature, annual rainfall, and altitude have been considered as environment conditions. And other thing is they have considered the management systems of each and every zone. That means intensive management system or semi-intensive or extensive management system. And thirdly, the suitable breeds for these environments and the blood levels of recommended breeds. For instance, for upcountry, they have recommended 100% European blood level, and we come to mid-country and dry zone, the European blood level has been decreased gradually. So that those are things considered. Thank you.
Thank you very much, sir. So we have questions on the audience. Dr. Chandika Kajagiri, I think, from
hello, and first of all, I would like to thank you for organizing this interesting forum, and especially for Animal Science Society for organizing this very interesting session. And also, I want to thank for all the speakers for their nice presentation and sharing their knowledge and experience with us. So I just want to ask the simple question from Dr. Seneviratne, that he discussed about the breeding policy. He mentioned that what are the factors they consider. Yeah, that through actually that we know that we have comprehensive breeding policy and well-written comprehensive breeding policy. The problem is that when it comes to the application level, field level, we can see that huge gap there with the policy recommendation and the field application. So that I just want to ask from the authority. So what is the mechanism that authority have to ensure the implementation of the policy at the field level? Oh, on the other hand, that if they violate or if they're not following the breeding policy, do we have any monitoring mechanism? So that's just I want to ask from the Dr. Seneviratne.
Yes, thank you for the question. First thing, I have to tell you that the breeding policy is not a thing that passed in the parliament as a act or something. So we have no authority to, there's no legal power for this this document. This is just a guideline. So somebody, if violate it, we can't do anything. In fact, since it's just a guideline, right? Okay. Other thing, to monitor this, we have planned distribution mechanism of semen. So we are handling the semen distribution mechanism. So we provide semen according to the breeding policy to each and every province. So that way we are trying to control the misuse of semen at field level. As I mentioned earlier also, this guideline has no any legal power. So we do not have take any action against the violating the breeding policy recommendations.
Okay, thank you.
Okay.
Thank you, Dr. Seneviratne and Dr. Chandima. And I have one more question that is related with the poultry genetics. But I think this question is related with all the other the livestock as well. Because the question is how the environment factors. We know the environment is playing major roles in animals' the productions and then the and other traits as well. So then how the environment factors can cause the genetics improvements of the whole trees. I think Professor Lee. Professor Lee. Professor.
Yeah. As far as I understand, the environmental factors is also very important because the animals face different environment. But for the breeding purpose, we set the environment is constant. So even though environmental factors affect the genetic trait itself, but we disregard the environmental factors. So if you consider environmental factors is too diverse, so we only focus on the genetic factors. That's my question. Is that okay for the answers?
Yes, Professor. So as we know that the G by E interactions is very much important for the production, right? So in case of selections of the animals, we have to consider the environment factors.
Yeah, that's right. Yeah, that's
for
right.
the poultry, but as well, but for the cattle and other the livestock species as well. So by doing the research on the how that the particular environments or the different environment in Sri Lankan case also very much important, because that's give kind of idea how the different the ecotypes behave in different environments. So then we can develop kind of the strains for the resources we have in different environments. So, but we have to do kind of research in that area.
Yeah. G by E interaction is also very important, but before you go to the G by E, you have to think about the non-additive genetic factors, which is dominance and epistasis genetic factors. So they are also very important because when you crossbreed different two diverse breed, making crossbreed, then non-additive genetic factors is also very important. So you better calculate the non-additive genetic factors and also the G by E interaction as well. So that's my suggestion.
Thank you very much. So there's one question from the audience. So it's also kind of improvements. Cattle. The question is, has there been any improvements in the cattle breeds in Sri Lanka, which produce the more at the low feed cost? So since we talking about the nutrition of the cattle, so the question is related with the nutrition and whether there is any the developed or improved breeds that thrive under the local nutrition conditions. I think Professor Indranil Pathare. Sir.
Yeah. I also can answer maybe, but maybe Professor Pradeepa has maybe a better solution. But anyway, as far as I understand that, I don't think that in Sri Lanka we do have a good, dairy breed developed for the kind of situation in Sri Lanka, as far as I understand. Although we have some tropical blood and when we produce, when we provide enough nutrition and the related environment, yeah, we can get enough. But as far as I understand, we do have some kind of breeding policy. But I mean, if it is successful at the moment, we should have our own breed. I mean, to have, with dairy. But at the moment, we do not have, I mean, that kind of breed develop in Sri Lanka to get the, potential within Sri Lanka and also to suit Sri Lankan conditions. We do have some breeding programs, and we have some temperate process in Sri Lanka, and we have imported some. Siemens continuously imported like AMZ, but in Sri Lanka, I don't believe that. Maybe Pradeepa madam can maybe answer better.
Okay, thank you very much, sir. So I think the Pradeepa madam was not in the panel, so I think I would like to ask some suggestion from Professor Kodituaku, sir.
Yeah, so actually it shouldn't be the target of, making a breed ideal to our climatic condition. If we are to self-sufficient the milk production, if we try to make a breed of our own that, thrive on our, low, whatever the resource input condition may not be the way forward. Of course, we can harness the things like disease resistant, maybe the temperate tolerant, but you need to incorporate those characters into the high producing characters. But like I saw one more question asking whether it's good to have high producing cow. We have to have high producing cow because our land extent is not getting extended. It's getting reduced day by day. So only option is we have to produce more milk per whatever the unit area that we have to do the milk production, right? So we need high producing cows, of course, through the breeding means, by incorporating, selection criteria, you can easily get in this, resistivity gene like temperature tolerance or disease tolerance or whatever. But of course, we should not reduce, or we should not lose the high producing capacity of such if we are going to make. So other thing is actually the mismanagement of nutrient. There are certain little things we can still do. Still, we have in our judgment, like last 5 years, we have been extensively doing this dairy project. So in our findings, what we found is we have enough potential, but the thing is the resource input level and the mismanagement of some of the things. Like, for example, the best example is the negative energy, the transition cow. We don't have such programs running even in large scale farms. So I think we need to find solution for that. And then even the existing, genetic makeup within our herd is even enough to go for self-sufficiency, even if we have, but nutrition is the key. So I don't know, even Saudi Arabia, they produce more milk than they require and they export. They don't even grow a single, pasture land. They just have very little, everything is imported. So that may be one option. I don't know. You have to do a cost benefit analysis, whether to put more lands into pasture, whether it's easier or cheaper to import from like Vietnam. I know some of the large companies, they started importing like rye, even the hay from countries like Vietnam, Cambodia, which was cheaper, than producing them in the country, so that they can produce the milk in cheaper price even than growing them in the country. So that's why we need to have a multidisciplinary approach. It is not just one discipline can decide this. It involves a lot. Economies are involved, sociology sometimes involved, genetics involved, like biology, veterinary medicine, everything. So it has to be something like a very holistic approach. Just importing animal would not support or would not help us to achieve this. All over the world, if you look at how they reach the self-sufficient in Asian countries, even in European, even in American countries, or maybe Canada or US, so they have this multidisciplinary approach of for finding the solution. So that may be the thing we are missing. The policy. The second thing. We don't have fixed policies. It just like the DAP as in only guidelines. There's no such fixed policies in terms of, especially in animal production in most of the cases. So people are even powerless sometimes, the administrative people, to stop certain things. So that's why it has to be a very, very, approach, I would say. Yeah. Thank you.
Thank you very much, sir.
Yeah, Prabuddha, I mean, can I add something to the same thing?
Sure.
Yeah, maybe we are referring to the same comment, maybe probably from one of the students, I guess. Actually, yeah, it's great to, have, students I'm thinking in this way. Yeah, so if I get the question, whole picture, the total picture, total picture, yeah, we have a power cut here, but still I can continue. Yeah, so the thing is that, for an example, if definitely, as Professor Suranga very correctly mentioned, that we should have high producing animals. So land is limited. Only thing is that if we correctly analyze the point, which is the point of the student. So definitely we should have a plan to feed these animals. For an example, that imported animals from Australia very recently, nearly 3-4 years ago, the project is completely failed because we didn't have a plan to feed those animals. One thing. The other thing was that the environment, we tried to manage these animals. So for an example, some of these medium scale and to large scale businessmen who didn't have experience in livestock industry had 200, 300 animals weighing nearly 500, maybe 400 to 500, at least around 350 to 450 kilogram body weight animals. So then he or she didn't have a plan to feed these animals. So although we are having high producing animals, as this student correctly understood, we should have a feeding plan first. So then we should have a potential and capacity to feed these animals. So then we can get the maximum potential out of these high producing animals. So other than that, so I believe, I thoroughly believe, in my personal opinion, we should select animals relatively, producing well. We have animals producing 8-10 liters and also more than 15 liters under feed levels, not just imported animals. So we can select these animals. So we know that we do have this kind of animals. So other than that, so it will be a real burden for these people who are involved in this livestock if we do not think about the potential, the capacity of the farmer who can manage this animal. Always we know that these farmers, they do like to have an imported animal at their farm. We very recently we had kind of experience with one of these projects came to this area with the government, and they are going to these people, they request animals from the government. They have never experienced livestock. Farmers who are already involved in this industry are struggling. So without, they cannot feed these animals. So in my point that before having a better animal, high producing animal, he or she, the farmer, should have the capacity and a plan to feed these animals. So then he can solve everything, and also the industry people and also the researchers, we can help them, and the channel, government channel, should take this to the ground. I believe, this kind of forum, I extend, I request, maybe no matter whether Uvavella is taking the leading role, we can get together, all these faculties and all these government channels can get together and have a common forum to, I mean, discuss these things. I mean, we cannot, I mean, forget these real field level things and the channels, and we cannot, this develop this industry without thinking about the, I mean, problems of real farmers. Thank you.
Thank you very much, sir. So I think I got several questions regarding the reproduction technology applications and AI related questions. So let me break the questions from the audience. So there's questions of what are the measures and the barriers that we need to take to popularize the artificial inseminations in the field levels. Also the why there's many abortions or any genetic relationship with associated with these abortions in the field level. I think, and also the question also related with the success of the AI. Especially at the farms level.
Yeah, can I get in again?
Yes, sir.
Yeah, okay. Actually, although we have already experienced this AI technology nearly now, nearly 8 years, right? I'm definitely around 70 years. So still we are struggling with a success rate of maybe around 30%. Actually, the thing is that, again, I'm coming to the same story. I mean, the government channel should have, I mean, from the farmer's point of view, they should have a kind of belief on this technique because that is successful. And we can list many issues to claim that we have problems to make this technique a success. I mean, starting from infrastructure issues and also problems with animals, and also the government, maybe the channel, and the farmers' knowledge on detection estrus. We can list many. But as far as I understand, even in this in farms, government farms, still, we suffer this. I mean, with this. So I don't think that, in my point, maybe Mr. Sunil Gamage maybe correctly mentioned, maybe in the same line. I mean, field application are really important to this overcome this issue. And I know that he is having much experience in this department. So important thing is, although we say that we practice this for a long period of time or decades, what is the extension work and the follow-up work we do at farmer level? I mean, for example, we cannot just think of blanket solutions. I mean, my suggestion is to have follow-up work at farmer level, kind of customized to each and every farmer. We do have enough officers in these, government officers. I recently visited this Kottapola, DS office. There are many people who, involved in this, industry. Only thing is they should train, we should train them well and should do a follow-up level, follow-up work at farmer level. Kind of customize follow-up, not just a blanket solution, in my opinion. That is my personal opinion, actually.
Thank you very much, sir. So there's one question, I think it's related with genomic selections using the high-tech, the Smp panels or the persistent selection. But the question is why we still going for the traditional selections method than using advanced method like marker system selection. So
I think I may answer that question.
yes.
Marker selections has been developed more than 20 years, but still not many markers available commercially. So traditional selection, for example, if you like the bigger cattle based on the weight, body weight, then all the possible positive markers you can select is just a selection of the looking at the body weight. So sometimes it's very difficult, even though the technology is very advanced, then still you have to think more about how can it's applied in the field. So very expensive technology is still available, but it's not very useful it's not covered everything for the animal industry. So that's why most of the countries still using the traditional selection method. Right.
You are correct, Dr. Lee. So I think I will do one share. There's a raise hand or Sunil Gamge, sir.
About selection, I think the Department of Animal Production and Health or the Agriculture Department before that started artificial insemination in 1950s. And we have a very good base population here of crossbred cattle on the one hand. On the other hand, we have a very good population of local cattle, which is kept by herders. Now, the Department of Animal Production and Health has, since 2010, have been collecting information, and they have a vast collected information on this area, Dr. Seniratna's branch, and that can be used for selection in different areas. That is one aspect of selection. We won't go into the type of selection we did in Canada or what we are doing in Australia or anywhere. We just simple, because we have all size values, progeny values, everything is available there. So that's one. Number two is why we are talking about increasing milk production. Why not we talk about using farmers, improving farmers' sustainability through milk production and increasing milk production? Because only now, I mentioned in 2001 census, we had 20% farmers with crop dairy integrated farming. Now we only have 15%. And out of these 100% farmers, 82% farmers are part-time farmers. So why, and because of that, now when this organic fertilizer thing came, they had no time to go and prepare organic fertilizer. One thing is they didn't have cattle. Other thing is they are working 12 hours outside and they are part-time farmers. And so why not we improve the sustainability of our project you will us using all this data, starting a project in that area, improving the sustainability of farmer through cattle farming, because our main thing is rice production. That is what we need our rice. We can't import, we don't have money to import. We need our vegetables, we need our fruits. And the farmer, if we are getting the farmer away from farming to part-time farming, that means these things won't happen properly. So why not we change our approach into using dairy cattle and indigenous chicken or whatever low input, because what I have observed is dairy cattle, they are in the dry zone now, all low input. They produce 8-10 liters with just morning they milk, then they go out, graze, come back and give milk again, you get 8 liters of milk. So, I mean, it's all almost nothing. So what we have to do is to breed using that information, breed an animal that can do the same thing, go out and graze and come back, and without getting corn from USA and maize from some soya from somewhere else, we give little bit of grains that we throw, and then get 10 to 15 liters. Instead of 15% farmers, we make it 30% farmers. Even with these 15% farmers, we produce 40% of our milk. If we really change management, this 15% can give 65% of the milk. So our problem is increasing milk production. We have to stop importing milk. We don't have money to import milk. Actually, that's the reality. So why not we look at it in a different approach? Instead of going into giving the best animal to give 50 liters, all the semen that we import from outside give 50 or 40 to 50 liters of milk, but when they send it to the field, they give only 8 to 10 liters. That's a genotype environment interaction. So my point is, why not change our approach? Thank you.
Thank you very much, sir. Your suggestions are highly welcome. And so with this one, so I think I would like to hand over the panel's the sessions to the by marking the end of the panel discussions to continue the rest of the works of the forum. And I would like to thank all the panel, distinguished panel members who joined today and gave their experience and knowledge and share their knowledge with us to improve the Sri Lankan farm animal breeding and reproductions to fulfill the future the demands we expecting. Thank you very much.
Yes, as Dr. Prabhuda Manjula mentioned, since we have reached the due time for the panel discussion, we will now take the session forward. We take this opportunity to thank the distinguished resource persons for their valuable time, and especially Dr. Prabhuda Manjula for chairing the event.
It is now high time that we announce and appreciate the extraordinary efforts of the participants of the logo competition held with the aim of boosting the creative thinking of department undergraduates and designating an official logo for the Farm Animal Breeding and Reproduction Forum of Ovulation University.
Yes, Rasika, the creative work of the participants, namely HML Sandipana, third year undergraduate, LPK Madhushankar, second year undergraduate, and WAN Neranjana, first year undergraduate, is greatly appreciated. And the logo designed by HML Sandipana, third year undergraduate of the Animal Science degree program, was chosen and will be the official logo for the annual Farm Animal Breeding and Reproduction Forum in the coming years.
We have now come to the end of today's proceedings. I'm privileged to invite Dr. Sudhini Fernando, Coordinator of Farm Animal Breeding and Reproduction Forum 2022 and Senior Lecturer, Department of Animal Science, to make the closing remarks and deliver the vote of thanks.
Thank you. Honorable Vice Chancellor, distinguished speakers, valued invitees from universities, industries of government and non-government organizations, deans of the faculties, heads of the departments, professors, senior lecturers, dear students, ladies and gentlemen. It's a great honor and privilege for me to deliver the vote of thanks at this event, the Farm Animal Breeding and Reproduction Forum 2022. I, on behalf of the Farm Animal Breeding and Reproduction Forum 2022, would like to extend my heartiest gratitude to guest speakers for sharing their knowledge and experience with us. Professor Pradeepa Silva, Senior Professor, Department of Animal Science, University of Peradeniya. Professor Jun Hong Lee, Chunnam National University, South Korea. Professor Suranga Kodituaku, Department of Animal Science, University of Peradeniya. Professor Indunil Patirana, Department of Animal Science, University of Ruhuna. Dr. P.G. Seneviratna, Director of Breeding Division, Department of Animal Production and Health, Sri Lanka. Dr. Hiruni Vijayasena from USDA Meat Animal Research Center, USA. So without your presence, we would not be able to have this fruitful forum, Farm Animal Breeding and Reproduction 2022. Further, we are grateful to our Vice Chancellor, Professor Jantalal Ratnasekara, for his continuous guidance and assistance throughout the event. I may like to extend my sincere thanks to Professor Saman Herath, Dean, Faculty of Animal Science and Export Agriculture, and Dr. Deshani Mudan Nayaka, Head, Department of Animal Science, Uvavelas University, for their valuable guidance and motivation to make this event a reality today. I also would like to extend my sincere appreciation to all academic staff members of Faculty of Animal Science and Export Agriculture for their continuous support. Well, the event like this cannot happen overnight. A big thank goes to Organizing Committee of Farm Animal Breeding and Reproduction Forum 2022, Professor Dinesh Jayasena, Mr. Sharit Dias, Mr. Shan Navaratna, Mr. Shamod Bashita, and all the academic staff members of Department of Animal Science for their enormous contribution in organizing this event. Further, the financial assistance extended by the AHEAD LTAC World Bank project is highly appreciated. Thank you, Professor Ruan Ranasinghe, Director OTS, AHEAD ELTA World Bank project, and Mr. Nuwan Lienege, Project Coordinator, and his team, Department of Animal Science, Uvavelas University, for their support. Also, I would like to extend my heartiest gratitude to Ms. Harshini Vikramaratne, Head, Department of Computer Science and Informatics, Uvavelas University, for permitting us to use Computer Science Laboratory, and Mr. Tisara Pereira for arranging the logistics. I must thank the administrative and non-academic staff members for the support during the arrangement of this event. My special thank goes to the Animal Science Society for the cooperation in hosting logo competition and for all the beautiful graphic designs they have done. We are happy to have a team dedicated and hardworking. We have been fortunate to have a renowned identity of academics and undergraduates of different universities in Sri Lanka, the industry, government institutions, non-government organizations, UW alumni, and other enthusiasts. I'm very thankful for the entire participants of this event. Ladies and gentlemen, once again, thank you for being with us today, and it has been a great pleasure. Thank you. Have a pleasant day.
Thank you, Madam, for your sincere words. Dr. Sujini Fernando, Dr. Prabhuddha Manjulan, your unwavering efforts to bring the first ever Farm Animal Breeding and Reproduction Forum of Uvavelas University to a remarkable and successful end are highly valued. Pramisha?
Yes, Rasika, this well-rounded discussion has undoubtedly provided us with insights that pose the present status of farm animal breeding and reproduction in Sri Lanka, highlighting the latest advanced techniques in farm animal breeding and reproduction from around the world, and the potential for improving farm animal breeding and reproduction in Sri Lanka to sustain the growth of the livestock and poultry sector in Sri Lanka. As stakeholders lodged at different levels of the industry, we are obliged to apply today's recommendations as more customized initiatives that come with a higher impact on developing the livestock and poultry sector of the country in short and long terms. Wishing you all a wonderful week and safe time home. Thank you.